Good afternoon, everybody. Good afternoon, everybody.
Good afternoon.
Recording in
Okay, it is now 1 o'clock. I'm going to go ahead and get started for the sake of getting through this. I don't know if we've had a chance to get through everything that was from the previous week, but because you have PowerPoints, I'm going to go ahead and say that we're going to go ahead and try to stay up with what it says in the weekly module. So I'm going to go ahead and start with 10A and 10B. And try to get through everything that's in the module. And then if we have time at the end and people want to go over something from the previous week, we can. We're also going to be doing a review at the end of today for exam 2. I can also spend a little bit of time with anybody who wants to go through the first exam question by question, but I won't have a huge amount of time for that, depending on how much time you guys have to spend with it. So I'm going to go ahead and get started. We're going to start with chapter 10A. And hopefully you guys have your notes printed out. Oh, you know what? I forgot to fix that chapter 14.
I thought we were doing 17A and 17B today. That's what you said last week.
That was from last week, but the problem was we don't have enough time to get through what we got this week and also do everything to set up for this week, as well as the test review and going over all the test questions. So anything that was from the past, I'm gonna let you guys just use the PowerPoints for, and then ask me at the end if you have any questions. That will also come up during the review, and I can lecture on any essays that will be coming up for this week's test having to do with those chapters. But I don't want to get behind again and owe the class something from the previous week. So I'm going to go ahead and leave that for now and do what is on the schedule for this week. And then if we have some time, we can go back. Okay, so I'm going to go ahead and share my screen. I think that I forgot to change. There was a incorrect link for one of the chapter 14. I think it linked to a PowerPoint instead of a set of notes. So I can do that today, by the way, while we're on, so that I don't forget. If I do forget, please send me another email and I'll make sure to get the chapter 14 lecture notes fixed. Okay, here we are at chapter 10A, muscles, and let's see. I think there was somebody who was trying to get in that, yeah, there's someone trying to get in. Okay, so let's do this again.
Oh, by the way, just to get clarity, is the exam 2 going to be on 17A and B?
It should say in the module.
Oh, does it?
Let's see. Yeah, it says right there in week 3 module exam 2, chapter 17A, 17B, and 12.
Oh, okay. Okay, thank you.
What we're covering today, unfortunately, doesn't go into this week's, but goes into next week's. So yeah, I will spend a little bit of time with 17 if we do have any time at all. Okay.
Can I ask a question real fast too, please? I'm sorry.
Sure.
When I go through the module, it doesn't give me discussion 3, but when I look under the to-do list, when I look at the calendar, discussion 3 is on there. Should we do it or leave it out for this week?
The only thing you ever have to worry about in this class is what's listed on the start here. So wait a minute, does it say there's a discussion? Yeah, so if it says no discussion board, don't do it.
Don't do it? Okay.
So use the week at a glance under the start here as your one and only guide for that. I need to get in. If you can send me an email reminder to move the due date on that. Sometimes it's hard for me to see what you guys are seeing. I only see what's in my start here. So yeah, please don't use the to do or what's due ever. Use only the week at a glance under start here, and that's the only thing you got to do. Okay, I got to keep moving. we'll do more questions at the end. Okay, so here we are, 10A, muscles. Okay, so muscles, and by the way, you guys see that the green is the stuff that you're filling in, right?
Yes.
Yeah.
Okay, eventually that's gonna run out, and you guys will have to figure out which words go into the blanks. But for these first few chapters, I thought you'd appreciate having them be color coded. Okay, so by the way, the only way a muscle can lengthen is if it relaxes and then something stretches it, either gravity. Okay, what was the last thing you heard me say?
Muscles tightening.
Okay, so the only way that muscles can lengthen is that they relax and something stretches them out, either an antagonist or opposite muscle or gravity. Okay, muscles, their primary job is to generate motion. They primarily move bones and visceral structures. The bones are moved by skeletal muscles. Visceral structures are always moved by smooth muscle or cardiac muscle if it's in the heart. They generate the same thing for the sake of understanding how this works. So force and tension are the same. Okay, muscles also generate heat when they contract. That's one of the reasons why you get overheated when you're exercising. You're generating a lot of heat. And so what happens is your blood moves through the muscles, picks up the extra heat, goes to your skin. The then cooled blood goes back into your muscles to cool it off, very much like the radiated your abdominal muscles support your abdominal cavity. And remember, I'm not going slow enough for you guys to fill it in as we go. I'm going to expect that you guys will either use the recording to fill it in or use the PowerPoint previously or after to fill it in. I need to keep moving through this. All right, different muscle types. Cardiac is found only in the heart. It's involuntary and it's autorhythmic. Autorhythmic means that it can contract without a stimulus. It's the only type of muscle that can contract all on its own without a stimulus. Smooth muscle is found in internal organs in the skin. It is also involuntary. Skeletal muscle is the only voluntary muscle. That means you can decide to contract it or not. Question. The muscle right under your lungs is called your diaphragm. What do you guys think? Is that skeletal, smooth, or cardiac muscle?
Skeletal.
Skeletal, yeah.
Okay, most people get that wrong. They say smooth. It is skeletal because you can hold your breath at will. And the only way to make a muscle stop working, or start working is if it's skeletal. Okay, so skeletal muscle attaches generally to bone. There are a few muscles in the face that don't. It moves appendages. That means arms and legs. It supports the body, especially in the back, upper back, and abdominal region. Antagonistic pairs work in opposition. For example, in your arm, you guys can see this is the biceps brachii and triceps brachii. Because they're on opposite sides of the arm, they're what are called an antagonistic pair working in opposition. Flexors. So in this case, the bicep would be the flexor because that decreases the angle. The triceps on the other side increases the angle. So flexors bring the angle inward, extensors increase the angle or basically stretch out the arm. Okay, so these are some images of what they look like. You have similar images in your book. Is a diagram, and the right side are actual micrographs. Okay, this is a picture from your text. What it shows you is what happens if you take an individual muscle apart. It's made up of a bunch of things called fascicles, which are bundles of muscle cells. Then within a fascicle, so if you take that fascicle or bundle, inside of it you see individual muscle cells. And then in an individual muscle cell or fiber, please make a note that a muscle fiber and a muscle cell is the same thing. So if I ask, is a muscle fiber the same thing as a muscle cell? Yes, it is. And so each of the muscle cells has within it these things called myofibrils. We'll get into that. Also notice mitochondria scattered throughout to generate ATP. All right. Skeletal muscle anatomy. It's about 40% of body weight depending on exercise level. For example, if you're a bodybuilder, it'd be quite a bit higher than that. And if you're a couch potato or obese, it'd be less. Myofibrils are the smallest contractile unit composed of both actin and myosin filaments. We're going to be talking about how those interact with each other. And there will be an essay on exam 3 asking you, how does how do actin and myosin interact with each other? So a muscle fiber is the same thing as a muscle cell. A fascicle is a bundle of fibers or a bundle of cells, as I showed you in the image. A motor unit is several fascicles that are stimulated by one motor nerve. So often it's the case that a single motor nerve will stimulate several fascicles within a muscle to contract together. Now, generally, not all the fascicles contract at the same time, and that's going to lead to a thing called recruitment, and I'll be talking about that later. A muscle is the anatomical unit, often or usually composed of many motor units, which would be a bunch of fascicles and a single motor nerve. The sheath surrounds fascicles and the whole muscle. And these sheaths converge to become tendons. So interestingly, when you see a tendon at the end of the muscle, it doesn't begin at the end of the muscle. Guys, if you have noise in the background, please mute yourself. They converge and become tendons so that the tendon doesn't actually end or begin at the beginning of the muscle. It goes throughout the entire muscle. And where you would call it a tendon at the end becomes the muscle, the sheaths around the fascicles or around the individual muscle cells within the center of the muscle. Okay, so this is showing you what a small section of the surface of a muscle cell looks like. This is from your OER text. And by the way, normally the outside of the cell is called a plasma lemma if it were a regular. But because it's on the outside of a muscle, you get the prefix sarco. So whenever you see that prefix sarco or the prefix myo, you guys might want to write that down. Sarco or myo, it's always talking about muscles specifically. So in a regular cell, you'd see an endoplasmic reticulum. Not so here, you have sarcoplasmic reticulum. This is going to be doing a very special job of releasing and absorbing calcium. I'll be talking about that later. The terminal cisternae are the end of the sarcoplasmic reticulum, and the T-tubule is an extension of the surface of the cell, extension of the sarcoplasm. And this triad, we're not going to worry as much about that, but the key is the interaction between the sarcolemma, the T-tubule, and the sarcoplasmic reticulum is going to lead to how muscles contract. Muscle fiber structure. Okay, multiple nuclei, and there's a blank in your notes that says why. So here's the answer. Muscle cells, especially skeletal muscle cells, are composed embryologically of several separate cells that end up fusing together to make a single, very long, thin cell. And the question is, why is that so important? Well, just to let you guys know, a muscle cell spans the entire length of the muscle. One cell goes that entire length. So for a muscle like your bicep, let's say 6 inches long or 5 inches long, that's an exceptionally long, thin cell. Now, why do you need multiple nuclei? Well, remember that the nucleus codes for the production of protein, and protein is especially important in muscle cells because the muscle is composed of protein. Those contractile structures of actin, myosin are protein. So you have to have a local nearby nucleus to control that protein synthesis as well as local activity in that part of the muscle. So having multiple nuclei across this very long, thin cell is very important because you can't have the nucleus too far away from any part of the cell unless it's an axon and a neuron, and we can talk about that later, okay? All right, the sarcolemma is the muscle cell membrane, as I pointed out in the image. The T-tubule, those are inward extensions of the cell membrane inside the muscle cell. Now, the reason why this is important is because of the fact that the action potential, which I really do need to spend some time at the end talking about how action potentials work because it is on this week's test. Anyway, so what it does is it allows the action potential that's coming from the motor nerve to spread across the surface of the cell and then to burrow into the center of the muscle cell so that all of the myofibrils that are associated with that cell can all be stimulated at exactly the same time. So even though muscle cells are fairly thin, you have to get the signal all the way to the center of the cell so that all the myofibrils can contract. So as I said, allows action potentials generated on the surface to reach the myofilaments even deep into the surface. The action potential is always going to be on the surface of the cell. The signal has to make it all the way to the center of the muscle cell. Why is this important? I just said. So it's to allow the signal to spread throughout the entire depth of the muscle cell so that all the actin myosin filaments can contract when stimulated. The sarcoplasmic reticulum. Now, normally in a regular cell, it would be responsible for either protein synthesis or lipid synthesis. But in a muscle cell, it has a completely different job. It surrounds each myofibrils and releases calcium for contraction. So one of its jobs is to release calcium to allow contraction to happen. And its other job is to absorb it when there is no. All right, the sarcoplasm is what we would normally call cytoplasm, but because it's in the muscle, it's called sarcoplasm. Lots of mitochondria are for ATP generation because you have to be able to generate a lot of ATP in active muscle. Glucose and glycogen are for energy. Glucose is simple sugar. Glycogen is a starch made up of several glucose molecules attached together. The myofibrils are the smallest contractile units. They're actin and myosin, and these are the contractile proteins that will interact with each other to allow you to flex your muscle. Okay, this is an image not from your book, but it does show how the sarcoplasmic reticulum in light blue interacts with the T tubules, bringing the signal inside. And then when the calcium gets released, causes an interaction between the actin, which is red, and the myosin, which is purple. Okay, this is an image from your book. It shows you that this is what a whole bunch of myosin fibers put together look like, with each of the heads sticking out. And this is an individual myosin molecule with the actin binding heads. Over here is actin showing its double twist here with the tropomyosin, which are the covers, and then the troponin, which they're not labeling here. The troponin is this tan thing. It's going to be a spot for calcium to bind, uncover the actin binding sites, and allow the two to interact and you to flex your muscle. Okay, actin are called the thin fibers. They articulate with myosin. And when they do that, usually what follows is a flexing of the myosin head to move your muscle. Myosin are the thick filaments. And by the way, in the previous figure, the actin was green, the myosin was purple. Titin. Okay, titin is a special material found in there. And what it does is allows two abilities or characteristics of the muscles. And what those are is elasticity, that is the ability to be stretched. And wait a minute, strike that, reverse it. Extensibility is the ability to be stretched. Extensibility. Then elasticity is the ability to snap back. Those don't always go together. We normally associate them, like a rubber band has extensibility and elasticity. But some things you can stretch out and they stay stretched out. For example, a piece of certain metals, stretch them out, they stay stretched. That's extensibility without elasticity. So extensibility is the ability to be stretched, and elasticity is the ability to recoil from that stretch. Most materials, when they have one, they have the other. Okay, the sarcomere. Now this is an image not from your book, but it shows you that the actin, which are red, surround each of the myosin bundles, which are purple. So you can see that many actins surround a single myosin, and several myosin surround individual actins. So it's a three-dimensional interaction. Okay, this is an image from your book, and this is likely to be an essay on test 3, not test 2, which is this week. So that can be a little bit confusing, but take notes here because this image will be on test 3, and I'll be asking you to explain it. So I'm going to take some time. I'm going to explain it, and my biggest recommendation for you is to rewatch this and take notes slowly. So you pause it, take notes. What I noticed on test 1 when I was looking at some of the essays, people were not watching the lecture, or they weren't taking notes on the lecture when I talked about it. They were going out on the internet and trying to listen to the lecture and repeat some of the stuff that I've talked about rather than just grabbing something off the internet. What was the last thing I said?
Most
people
noticed
go out on the internet and find several different sources and kind of mix it together like Frankenstein's monster, rather than having a cohesive narrative. Your best way to get the most points out of me is to listen to my lecture and relate some of the things I talked about without it being a transcript. So that's the best way to get it. Okay, here we go. So we're going to start at step 1. This is, notice that we have, well, this does it in a different order, but that's fine. Okay, so in the very first step, and by the way, this is a cycle, so you can start with any step you want, but in this case, I'm using a different text than I've been, so normally they would be starting it right here at D, but we'll go up to here. So what we're going to see here is the previous step. You guys see ATP down here? What has happened is that adenosine triphosphate, which is energy, has separated or dissociated. And you see the phosphate attached and the adenosine diphosphate here. Now, notice that the myosin is still attached to actin. And the reason why it's able to be attached is because these little gray circles, calcium, have bound to this molecule, which is called troponin. It's not labeled there, but it's called troponin, and you'll see it coming up in the notes. So what happens is when calcium binds to troponin, these tropomyosin, these kind of spirally brown covers to the actin filament, move out of the way, allowing the myosin to interact. So because there's calcium present, the myosin and actin can interact with each other. And you're going to want to say something about that because it shows the calcium right there. So we're beginning with adenosine diphosphate and the separate phosphate attached. Now to get to step 2, the flexing, notice it's straight here and flexed here. The ADP and phosphate have to leave. So when they leave, that initiates the power stroke. Everybody see that? And you can get more details about this in your text. But the actin across the surface, and that's really the fundamental business.
Can you repeat that last part? You had froze.
That initiates the power stroke, and the actin moves relative to the myosin. Now to get to step 4.
Professor, can you repeat that last part? You had froze.
All right. What initiates the power stroke is the release of the ADP and phosphate, and that allows the myosin head to flex and the actin moves across the surface of it, which is fundamentally the microscopic component of flexing your muscle. All right. Now moving on to D. Now, if you want, now, so what they don't show is this just sitting here attached to this. Now, here's a really important thing I want you guys to get. You notice that myosin is attached to actin. What allows it to dissociate, to detach, you see the gap right in here, you guys? The gap that allows it to let go is the attachment of another molecule of ATP. Now, I'm going to take a little side note here, and I want you guys to take notes. Have you guys ever heard of a thing called rigor mortis?
Yes.
Do you know what it is?
Yes.
Like, right. So when someone dies, after a certain amount of time, their body becomes rigid. I just caught a rat in a trap and its body was like frozen, even though it wasn't cold. That's rigor mortis. What happens is this, the reason why rigor mortis happens, and that's one of the ways that you can identify how long a body has been deceased, is right after somebody passes away, there's still plenty of ATP in the muscle. So the muscle remains loose. So if someone has just passed away, they don't immediately go into rigor mortis. But as soon as all the ATP has been used up, if there's no more ATP here in step D, myosin, the purple, and actin, the green, can't come apart. And so there's the rigidity. The actin and myosin are held together. So the muscles become rigid because you can't get from step C to step D without ATP attachment. And that's the key. And I want you to talk about that in your essay. And test 3 is without actively produced ATP, myosin can't release. Now the question is, do bodies stay rigid forever? No. What happens eventually is decomposition takes over and breaks apart this association between actin and myosin. So, but that's quite a bit later after rigor mortis. So if I ask you a question about rigor mortis, remember I talked about that happens due to the lack of ATP, allowing the myosin to dissociate from the actin. Okay? Now, the step that once ATP attaches, it lets go, but what causes it to be cocked and straight again and ready to attach to another actin green circle is the dissociation of ATP into ADP and phosphate. Notice that now we're back here. So as soon as that happens, it reattaches to another actin and repeats the process. Do you guys get how that works? So ATP allows it to let go. When it dissociates, it makes the myosin head become straight and then attach to the next actin molecule, and we're back to the top. So it's a cycle. You guys get it? I know it's a lot. It does talk about it in your text, and you can rewatch what I'm saying, and you can ask questions at the end if you want. We've got to move on. Okay, now we're moving from what's happening at the actin and myosin filaments to what is happening at the surface of the cell and moving down through the T tubules, etc. Okay, so a lot of this isn't labeled, so you guys are going to want to perhaps label this, even if it's not labeled in the figure. So I'm going to go through each of this, and by the way, this is going to be part of the essay as well. I'll probably have the previous figure and this side by side, and I'll ask you to tell me all about it. Now, interestingly, this is what happens first. This is what happens first, and there's a little bit. What's happening? What was the last thing you heard me say?
There's a little bit.
There's a little bit of a disconnect, because what they're showing happening here is the opposite of what's happening here. It's not a great figure. I had a much better figure in my previous text, but I wanted it to match what you guys have in your books. So what it means is you're going to have to take good notes that what's happening at the very top is kind of the opposite of what's happening right here. But if you take notes, you'll get it. Okay, now I did not lecture on how an action potential works, which I'm going to try to do at the end of today if we have time, which was chapter 17, which is this week's test. But so they're going to be a little backwards, but you guys are going to have to figure it out. So when we get to the end of the action potential, what we're going to see is a signal. The action potential is coming down. This is the end of an axon, which is the extension of a neuron. And if you guys have already filled in your notes and worked on the review questions, you should know what this is. Each of these little bubbles is called a vacuole, and it's full of acetylcholine or another neurotransmitter. I tend to focus on acetylcholine, abbreviated ACH. We're going to see that in 17 when we get to it. Now, when an action potential or nerve signal arrives at what is called the axon terminus or axon bulb, it causes exocytosis. And I talked to you guys about what exocytosis is, and that is what is on the inside of a bubble gets dumped to the outside. Now, these little red dots are the neurotransmitter, usually acetylcholine. When that binds to the surface of the cell, it starts opening sodium potassium routes, which we've already talked about quite a bit. And that causes the beginning of a muscle cell action potential. Now, before you guys see, this is a signal happening, and this is what's happening between signals. So I don't like this figure, but it's the only one that we have for your book. So when there is no signal, calcium is removed from the sarcoplasm or muscle cell cytoplasm that moves it off of these little tan circles. And that causes the tropomyosin, which are these brown strings, to cover the actin binding sites, preventing the myosin head from attaching. So this is basically, all of this is showing relaxation. But let's do the opposite. Let's talk about when an actual signal comes. The signal comes down here. You have exocytosis. This acetylcholine binds to these sodium potassium gates, opens it up, causes a signal, starts spreading. So instead of calcium being reabsorbed, it gets released. It goes over here to the tropomyosin and causes this to open up. Then the myosin attaches to the actin and you have flexing. So what they're showing, this whole bottom part is how you relax a muscle, but the top part is how you cause it to flex. Not my favorite. That's all that was available in this text. And I had to use it because it's free, right? Nobody wants to pay for books anymore. So I don't know what you guys are going to be able to do with it. Do your best. So the bottom half is how you relax a muscle, and moving all of these from this gap and shutting off the signal, then all this would be correct. But if you're having this acetylcholine come across the cleft and open up these channels, then the reverse happens. I hate to give it to you guys that way, but that's all we got. Okay, continuing on. Fiber contraction, speed, fast twitch. Now, you guys all know what dark meat and light meat is when it comes to chicken and turkeys, right?
Yeah.
Yes.
You guys know what okay.
Yes.
Now, humans don't have light meat and dark meat. They have combination muscles. The only place that you would see what you'd call light meat or fast twitch muscles is in the external eye muscle, the muscles that make your eye go up, down, left, and right. Otherwise, almost all the rest of our muscles have a combination of fast twitch and slow twitch. Now, the key is they're anaerobic and they fatigue easily, which means you can't hold it for very long. Now, think about all the muscles in your body. You could hold on to something with a moderate amount of force and hold it for quite a while. There's only one set of muscles that you can't hold for more than a few seconds, and it's your eye muscles. So if you guys want to see how that works, you can do this demonstration. Then look up and try to hold it. So don't put your head up, just put your eyes up, and ooh, that starts to hurt really quick. What did you guys hear me say?
You didn't cut out for me.
You can't hold your eyes open for very long.
No, it's not holding your eyes open. It's holding your eyes up.
Yeah,
So
in the
the idea is
position.
point your nose directly forward, but look up and try to hold it. You can only hold that for a few seconds. What it demonstrates is that the muscles that move your eyes don't pull and hold. So what normally happens when you look up, you lift your head and your eye goes back to normal. Does that make sense? You turn
Yes.
your head towards what you're looking at. The eyes are only supposed to move quickly with your head following. But if you try that experiment with your head forward and your eyes up, you can only hold it for a few seconds.
Okay,
because
that
that's
makes sense.
fast twitch. Okay. What do we use it for? Combination of slow twitch and fast twitch. But what they do is give lots of force. So whatever muscles are really high power muscles tend to have more anaerobic fast twitch muscle fibers in them. Fast or delicate stuff like your eyes, very fast moving muscles. Playing piano, that your muscles in your fingers and hands are very much a fast twitch. Now, the muscles in your forearms that are responsible for hanging on to something are a combination of fast twitch and slow twitch. Sprinting is another good example of that. Sprinting you can only do, we talked about this, you can only do it for 10 to 20 seconds maximum because you do switch pretty quickly into anaerobic respiration. Okay, so looking microscopically, you can tell now why dark meat is dark and light meat is light. Now, humans, I told you, have a combination of the two, but chickens and turkeys don't. So the question is, what do chickens, let's just take chickens for an example. What do chickens use their dark meat for? You guys know where you find dark meat on a chicken?
In the thighs, right?
Thighs and drumsticks, right? What movement would those things be doing?
Walking, running.
Walking. Do chickens spend most of their day walking around?
Yes.
Do chickens normally sprint
Very
Yeah.
short time frames.
for a very short time frame, right? Very short. If they really need to go fast, they tend to flap their wings. They don't sprint. So the idea is that the dark meat in the legs is for constant aerobic activity, right? Chickens are walking all day long. What about the white meat? Where do you find white meat in a chicken?
The breast.
The breast. Okay. How many chickens fly south for the winter?
None.
None.
The
They
joke.
don't.
They don't, because they have all light meat. And the reason is the following. Is remember I told you it gives very fast, very powerful contraction, but it's anaerobic. It can't last forever. So chickens and turkeys are both in the same kind of family. And that is birds that walk around all day. They don't fly all day. And then if something attacks them, they have to have enough power to lift this fairly fat bird up into a tree quickly, right? Think about how heavy turkeys are. You ever pick up a big one? They're heavy, right? Like like the one in the back. So if you don't have fast twitch muscles in the breast, it's not going to be able to lift itself up away from danger quickly enough to escape. Now, question. What kind of breast color would you expect to see in a goose or a duck or any other migratory?
Would
Dark.
it be dark meat? Would it be dark meat?
It'd be mostly dark meat with some light. I mean, you would see it kind of like a mix. It would be kind of like a light to get going, but then they also have to be able to maintain that for long periods of time. That's how geese are able to travel hundreds of miles in a day, because their breast, instead of being light meat like a chicken, is mostly dark meat with a little bit of light in there so that they can fly for long periods of time. Now notice that ducks and geese can't just jump right up in the air like a chicken can. They can't get right up into a tree. They have to flap their wings for a long period of time and get going and then take off, right? That's because they don't have the huge numbers of anaerobic fast twitch muscle fibers. So hopefully that will help you understand the difference. All right.
Could humans that are marathon runners that get in like 30 miles a day, could they?
Yeah, humans. So that's actually a good point. Weightlifters, power lifters tend to accumulate more fast twitch muscle fibers, and marathon runners tend to accumulate more of the darker slow twitch. So good point. It depends on what you exercise for. If you're doing a lot of really powerful short-term things like weightlifting, then you would have more of the light twitch fibers in them, or the fast twitch lighter colored ones, and marathon runners would tend to have more of the slow twitch aerobic. So good point. Yeah, you can actually modify your muscles depending on exercise. Okay, so here we have the slow twitch. This would be the dark meat or dark fibers, less fatigue. There's lots more mitochondria. The question is why? Well, remember, mitochondria have to be able to generate ATP. And if you're not going to fatigue, it means you have to continue to supply ATP without going into anaerobic respiration that we talked about earlier in class. More capillaries gives more oxygen, so you can constantly be supplying the muscle cells with ATP using oxidative phosphorylation or aerobic respiration. Myoglobin. Okay, let's talk really quickly about myoglobin. You guys have probably heard of hemoglobin, which is the red pigment in red blood cells that allows it to pick up oxygen at the lungs. Well, here's something interesting. Myoglobin is found in the slow twitch muscle cells, and it has an even higher affinity or attraction for oxygen than does hemoglobin. So what that means is that even though the hemoglobin is holding on to the oxygen, the myoglobin will pull it from the blood into the slow twitch muscle cells and making sure it constantly is being delivered. So marathon runners, geese, migrating birds are going to have lots of myoglobin, lots of capillaries in their muscles, lots of mitochondria, darker meat.
So does that mean you would find more iron in the myoglobin? Because the iron is what holds on to the oxygen.
Myoglobin does have iron. So yeah, you would see that's part of the reason why there's darker color, you guys, is because the myoglobin has this kind of dark brick red color. And that's why dark meat looks dark, because there's lots more myoglobin in the dark meat. Does that make sense? And that's part of the reason why dark meat is actually more nutritious, because it has more of the stuff. I mean, it doesn't have as much, it's not as packed with protein as the white meat, but you are going to get more nutrients in there because of the kinds of activities that muscle cell does.
That makes sense.
So back to slow twitch. So endurance activities. So the muscles associated with walking, especially postural muscles, your lower back and some of your upper back and your neck, all the things that have to be holding you all day. Now, why does your back have to hold you all day? Well, your spinal cord or your vertebral column is on the back of you, not in the center of you. It's on the back. That means the weight of your body is on the front side of it. And that means it wants to crumple forward, right? And so all those muscles on your neck, back, and lower back hold you upright, even though most of the weight of your body is out in front. So without those postural muscles, gravity would pull you forward. And that's what happens when those muscles relax and somebody falls asleep, they slump forward. Okay, so the postural muscles especially are the slow twitch, darker colored fibers. Okay, there's a length tension relationship. This is not from your text, but the more cross bridges you have, the more tension. When we say cross bridges, we mean interaction between actin and myosin. In this case, the myosin is green, the actin is purple. Sorry for the switch there. But what this graph shows you is that at a particular muscle length, you're going to have the greatest amount of overlap here. And when you start to overlap them too much, like my fingers, see how my fingers are starting to overlap too much and there's no more room for them to shorten against each other. So you're going to lose power there. And then when my fingers like this, there's not very much overlap at all, less power. So when your muscle's at a medium length, that would be your arm at about an intermediate angle. You're going to have the most overlap between actin and myosin and the most power. Okay, that was, where were we, 10A?
Yes.
So we're gonna do 10B, and then we're gonna jump back to 17 and get caught up. I just don't wanna be,
Get
we
behind.
have 14 to do. You know what I may end up doing? Because I do realize that there was a problem with the chapter 14 actual lecture notes. I may punt on 14 to next week. I do really want to get through 17 because you guys do have a test on it this week. So I'm going to finish 10B. We'll go back to 17 and then do our review for the test. And so even though I said we're not going to get behind, I guess we're going to get behind. All right. Where is my mouse? I don't see my mouse.
Will you be covering chapter 12? Just 17?
Did we miss 12 as well? How many
Isn't 17 and
chapters are
12
we
for,
behind?
for this coming exam?
Yes, we haven't gone over neither of them.
Oh my gosh, we're behind. Okay, I'm gonna do 10B and then we'll jump back to 12 and 17. And whoever wants to hang out can hang out. And then we will we'll do our review at the end. If you guys really want to do a review before we get completely caught up, we can do that too. But for those people who want to go over exam 1 with me, the questions that they missed, that is going to be for the very end. So yeah, too much stuff to do, too little time. Anyway, so here we're going to go with chapter 10B, and then we'll jump back to 12. So at least we won't be completely behind next week. We'll only be partly behind. All right, muscles. What is a motor unit? Now, I talked about this a little bit earlier today when we were talking about 10A, and that is all of the muscle fascicles. And in this picture, you can see each of these fascicles is a different color, or they're color coded. So this is motor unit 1.
We can't see anything.
Yeah, we can't see the picture. We only see who's on this.
Wait.
Zoom.
Maybe I didn't do share screen. All right. Can you see it now?
Yes.
Yes.
All right. Okay. Sorry, guys. Playing catch up sucks. All right. Each of these colors represents a motor unit. That is all of the muscle fascicles that are going to contract when this particular, in this case, this red neuron, color-coded red, sends a signal. Then there's blue and there's purple. Now, what we mean by all or none, which means that when there's a signal coming from one of these neurons, all of the muscle fibers within the fascicle are going to contract together, kind of depending on how strong the signal is. But for the sake of your understanding of this, it's an all or none prospect, which means if a signal arrives, then all of the muscle cells that are attached there are going to at least begin to contract. Now, what we mean by a low ratio, fine touch, for example, your finger muscles and the muscles of the external eye, it means that you have about an individual nerve fiber to 6 ratio. 1 nerve, 6 fascicles. But with fine touch, like your fingers that give you lots of control, or your extrinsic eye muscles. What was the last thing you heard me say?
Intrinsic eye muscle.
Intrinsic eye muscles. There's going to be a 1-to-1 ratio. That means for every 1 nerve fiber, there's going to be 1 fascicle. And what that does is it gives a lot more control over those muscles, not as much power. When you have okay, so the finger muscles and extrinsic eye muscles, but big muscles have a high ratio. What that means is, for example, the most powerful muscle in the body, believe it or not, is your gluteus maximus. So in your gluteus maximus, there would be 1 of these neurons and dozens, if not hundreds, of fascicles associated with that single motor unit, with that motor neuron. Why is that? Because we generally don't need to do fine touch things with our buttock muscles. That's for running, walking, which is kind of a more less fine touch sort of thing. So if you need to be able to have a fine motor skill, you're going to have a lower ratio. And if you want a power muscle like the quadriceps or the buttocks or the biceps brachii, you're going to have a high ratio. That means 1 neuron to many fascia. So in some cases, it could be 1 to 2,000. 1 motor neuron to 2,000 fibers. So leg muscles and the larger muscles of the arm and shoulder would have a high ratio. Okay, what is recruitment? So in a weak stimulus, let's say a smaller stimulus is coming through this neuron, only the lowest threshold fibers contract. What do we mean by that? Now, within an individual fascicle, there's going to be some of the fibers or cells that are more sensitive to a very, very small signal level. So only the lowest threshold fibers are going to contract, and that means your muscle's not going to pull quite as hard. It's going to be for a softer contraction. These are going to be slow twitch muscles, typically. When there's a high stimulus, that means lots of action potentials coming through a particular neuron. All to maintain a contraction for long periods of time if you're contracting all of them at the same time. Well, you can't. So if you ever think about what's the most that you could lift with, let's say, your bicep, the greatest amount of weight you can. I can't hold that very long. But what if I lift 20? I can hold that a lot longer. So why is it that even though each than 50, and it's because of this phenomenon that you see right in front of you. So what's going to happen with a less than maximal amount of weight, these motor units take turns. So for example, the red will pull for a while. When they get tired, the blue takes over and it starts to pull. Now, while the blue is pulling, the red is resting. When the blue gets tired, the purple takes over, and then both the other ones are resting. And then what that allows you to do is to pull and hold for long periods of time, even though each individual bundle is going to become exhausted at the same rate. So it determines how many of these motor units are going to be recruited on how long you can maintain the contraction. So if it's a maximal contraction, you recruit all the motor units and you get quick exhaustion. But if you have a much less one, you take more time between the motor units and you have a lot more endurance for that activity. So the motor units take turns during asynchronous contraction. And the analogy that I like to use is, could one person effectively carry another person of the same size out of the desert by themselves? And the answer is no, without some kind of a stretcher or something like that. You get too tired holding them on your back. But if you had 5 people, one person could carry the person on their back for half an hour, give it to somebody else, and then rest as they walk. And if you had people taking turns carrying the body or the person who's passed out or whatever, then you could get the person effectively out of the desert and nobody would become exhausted because only one person at a time is doing the work, the hard work. And that's what's going on. Where you contract a muscle for a long period of time, it's because these motor units are taking turns. And the more time they get between contraction, the longer that you can hold that contraction overall. Okay, joints. All right, joints. Tendons attach muscle to bone. And I did mention to you guys that tendons go throughout the entire muscle, making up the connective tissue wrappings within the muscle. Ligaments attach bone to bone. Now, when I ask people which one has to be more flexible, they tend to say tendons, but it's not true. Ligaments are the flexible. Your joints. The fact that you're able to bend your wrist and bend your ankle is due to the fact that the ligaments that are attaching bone to bone are flexible, more like rubber, whereas tendons tend to be more like cable. That attaches muscle to bone. Stronger, less flexible. Ligaments, bone to bone connection, more flexible. Muscles generate the contraction force. Now, we're going to talk about some different types of contraction. Isotonic is when something is moved. So imagine that you pick up something that weighs 5 pounds. I'm guessing most of you guys can lift that. Now, the reason why they call it isotonic that weighs 5 pounds in your hand and it's being lifted up.
You froze again, right after you said isotonic.
Okay. The reason why they call it isotonic is because iso means the same, tonic means tension. So for example, if you had a 5 pound weight in your hand, most people can lift that. As it's being lifted, the maximum tension that the muscle can produce is 5 pounds. If you pull any harder, it just moves faster. If you have a 5 pound weight, maximum tension that can be created by the muscle lifting it is 5 pounds. So if you are moving it, it's called isotonic because it means same tension. Now, if you don't have movement, it's called isometric. Iso means same, metric means length. So for example, if I was trying to lift up something that weighed 300 pounds, I can't lift it, but I can pull and I can pull and I can pull harder and harder and harder up to a maximum, and then I have to relax. So in this case, my muscle doesn't shorten, it remains the same length, but the tension gets greater and greater and greater up to a maximum before I give up. Does that make sense? Isotonic, same tension if you're moving it. Isometric, same length, no movement, but you're going to increase the force up to a maximum. Remains constant throughout the shortening process. During isometric contraction, exerted force does not cause the load to move and length of fiber remains constant, even though you're continually adding more and more force. Now, slightly different thing is eccentric contraction. Have you ever set something gently onto a table? Well, you're not just relaxing the muscle and letting it fall and crash, you're setting it down gently. So for example, if you had a 5 pound weight, you would only exert 4.5 pounds of force, and that would allow you to set it down gently. That's called an eccentric contraction. And that's what we do when we're weightlifting. We do a isotonic contraction in our flex, and then an eccentric contraction to let it down slowly. Isometric is if it's too heavy and you can't lift it. Okay, so this is a picture that's not in your book, but it does show you how isotonic, the maximum tension that is available is 20 kilograms. So it goes up to 20 and then you can't generate any more. Now in isometric, because this is too heavy for this muscle, it goes up to a maximum and then diminishes, but you don't see any shortening of the muscle. Even though this muscle can generate more than 20 kilograms of force, and that's why this moves, this one can't lift 30, so it never does shorten, and this is the force that would be required. So isotonic, it moves. Isometric, it does not. Okay, twitch, summation, and tetanus. If a muscle is stimulated by an increasing frequency of electric shocks, its tension will increase to a maximum, what is called incomplete tetanus, which are these little bumps that you're going to see in your text and perhaps in one of your simulations. If the frequency is so fast that no relaxation occurs between stimulus, then it's called complete or fused tetanus. So that means the muscle is pulling and holding as hard as it can. If muscle is repeatedly stimulated with maximum voltage to produce individual twitches, the successive twitches get larger. This is because the calcium doesn't have a chance to be pulled back into the sarcoplasmic reticulum, and it starts to accumulate, meaning more tension, more cross bridges, more actin and myosin interacting with each other and flexing. This is called TREP, or the staircase phenomenon. So it goes up, the tension goes up, you stimulate again quickly, it's going to go up again and up again because you're stimulating it to release more calcium, but you don't have enough time to pull it back in between the contractions. So this is an accumulation of intracellular calcium plus 2. Okay, so this is an image. I don't believe this is in your book, but it does show it. So for example, you stimulate it at each of these spots here where it starts to go up. Because they're so close together, it starts to go down, but when you stimulate again, you release more calcium, it goes up again, up again, up again, and then when you stop, it drops off. But if you do it really, really fast, it goes up to a maximum tension, which we saw in the previous figure, until it becomes exhausted. Exhaustion then means you run out of ATP and it relaxes. So this is called TREP, the staircase phenomenon, as it goes up. This may be in your book. I think it might be in your book. So check it out. All right, smooth muscles. And by the way, here's one of the fill-ins way at the top. Normally I don't do fill-ins. Did I cut out? No, okay. Smooth muscles have what is called a homeostatic role. That makes sure that blood moves the way it's supposed to, food moves through your digestive tract. It means keeping things as they're supposed to. Now, this particular figure. This particular figure shows you how long most of these muscle cells contract. For example, skeletal muscle, especially fat twitch, tends to be very quick. Cardiac is similar with a fast twitch, but smooth muscle takes a lot longer to contract and a lot longer to relax. And so it's going to not be a really fast contraction. It's going to be more of a tonic contraction, which means a long-term contraction. Often that controls fluids, for example, blood or digestive juices through your body. Sphincters, that basically muscle tissue in a circle that controls entrances and exits to these tubes. And tonic means a long-term contraction. So you can see a smooth muscle takes a lot longer to contract. It can remain contracted longer with less energy, but it takes a lot longer to relax as well. It supports tubes like your esophagus, which connects your mouth to your stomach, and blood vessels. BV is blood vessels. It moves products through your gastrointestinal tract. It has slow contractions, very little fatigue, very little oxygen use. So you can see it's a very useful thing for having long-term contractions. More characteristics of smooth muscles. So to stimulate smooth muscles, they are electrically coupled. That means they're connected to each other. Skeletal muscle are not. Each skeletal muscle cell is completely separate from the other ones electrically. So cardiac muscle is also electrically coupled. Hormones may stimulate or mediate contractions. So this is one of the few muscle cell types that can use hormones to stimulate. Skeletal muscle is not that, and normally cardiac muscle is not that either. Only smooth muscle can be stimulated by hormones. The shape, they're pointed. So it's kind of like a stretched out football. You guys know what a football shape is? That is spindle shape, and it's really pointy on the ends. That's the shape of smooth muscles. What makes them different in the way they contract? Well, there's longer actin and myosin fibers, so there's fewer repeating units. So instead of actin, myosin, actin, myosin, actin, myosin as repeating units, it tends to be spirally or oriented or arranged, meaning more contraction. It allows greater contraction distance because instead of just these small little sections moving, you have these larger sections moving against each other, and that allows a greater contraction, total contraction number. I think I said 30% for skeletal muscle can go up to 40 or 50% contraction for a smooth muscle. As I said, the arrangement of actin myosin filaments are diagonally crossed, which allows them to have longer filaments on a shorter cell than you would find on skeletal muscle. So here's a picture of it. It shows you the diagonal arrangement by these lines. These lines are the actin myosin filaments. You see they're diagonally arranged. I believe this is from your text, and what connects them are what are called dense bodies. So this is a relaxed cell showing it stretched out, and then when they contract, they shorten this way. Okay, a little bit more information about smooth muscle contraction. I'm not going to go into a lot of detail here because we're running out of time, but study this, look at it for the test. Again, more information about smooth muscle. I'm not going to spend a lot of time lecturing on it, but look at it, spend some time with it, read about it in your book. Okay, smooth muscle has no sarcomeres. It does have gap junctions, which is something I'm going to be talking about next, back from chapter 12. It contains 16 times more actin than myosin, which is a greater ratio normally you would find in skeletal muscle. Allows greater stretching and contracting, and they're anchored to dense bodies. You can see the dense bodies here, and you saw them in the previous one, instead of to the Z lines and M lines that you would find in skeletal muscle. So there's that, showing you the dense bodies and the intermediate filaments, which are basically the actin myosin filaments. Okay, cardiac muscle. These are built completely different. So instead of skeletal muscle, which are very, very long, thin muscle cells, or pointy fat in the middle, pointy on the ends. In this case, you can see they're branched. They look like kind of like tree trunks, but kind of tree trunks that have been melded into each other. There's always a single nucleus per cell, as we also saw in smooth muscle. Remember, no multiple nuclei per cell like we saw in skeletal muscle. They have a thing called an intercalated discs, and you can see them right here between the two cells. So where you see this junction, this kind of wavy junction, is what is going to call an intercalated disc. What those allow is the signal to spread from one cell to the other in the same way that you would see in smooth muscle. How do you stimulate them? They use pacemakers. Now, a pacemaker is what everybody has, but when people have a pacemaker in their heart, It usually means an artificial pacemaker because the one that was built into you isn't working. But the SA, which is the main pacemaker, and also the AV node, we're going to be talking about that when we get to the heart, set the rhythm of the heart rate. Autonomic, not conscious control. So you can't make your heart stop for a second and then start up again the same way you can with breathing or your skeletal muscles. You can slow your heart rate down by relaxing, but it's not a direct control of the individual beats. That's why they call it autonomic. And that is the end of that chapter. So what I'm going to do now is, what is our first chapter that I did not cover from this week's test?
17A. Basically, all of chapter 17 and 12.
Okay, I'm going to go to 17A, you guys. So get out your my internet's not doing well today. I'm sorry, you guys. Sometimes it's good and sometimes it's bad. It's not even that windy here. I just have a crappy internet connection today. All right, so we're going to go to 17A. Please get that out. So we'll do as much as 17A, B, and 12 as we can. Let me fire that up. So this would be 17A. Endocrine. All right. Here we go. Share screen. Share. Go. Okay. Did somebody have something in chat? Hey guys, if there's noise in your background, please turn off your mics. Because if I hear like kids talking or whatever in the background, it's very introduction to the endocrine system. Off we go. The study of hormones and their functions is called endocrinology. You're welcome, Des. All right. Hormones work on their target cells in 3 basic ways. The first is to control rates of enzymatic reactions, to make enzymes go faster or slower. And that has to do with inhibitors, often the recruitment of inhibitors, I think that we already talked about. Control transport across the membrane, so endocytosis and exocytosis can be controlled by the endocrine hormones. And most importantly, what we're going to be talking about mostly today is controlling gene expression and protein synthesis. So how does the nucleus know to start producing a particular protein? Well, often it's because there's been a signal sent from an endocrine gland, either in the rest of your body or in your brain. And it tells your body, hey, we need more protein of a particular type. All right, overview. Now there's another kind of molecule called pheromones, and that is organism to organism. Now, you guys may have heard that when
you know, on the sheet, it has 2 statements that we're supposed to fill in that were not on your PowerPoint. It says peptide blank based hormones cannot enter target cells, surface receptors bind blank. Where are we supposed to fill that in?
There?
They're actually on the page twice. If you look farther down, about halfway under hormone receptors, they're on there again.
Oh, it's
Okay.
out. Oh, it's out of order. Okay.
Yeah, sorry, it ended up out of order. I'm trying to transition between 2 separate texts, and sometimes it's not as clear as it could be. So let's just go to pheromones, and then if we don't get your notes filled in, let me know. So, okay, so you guys all know that when a female dog goes into heat, often male dogs will show up. Well, how is it that the male dogs know that the female dog is in heat? Well, they give off what are called pheromones. And the same is true for moths, that where they need to mate with each other. They give off these, what are similar to hormones, but that fly through the air and are picked up by other animals and read as, hey, there's something happening with this other animal. And the question is, do humans have or use pheromones? Well, there is a question on the test that says, what is the problem with talking about human pheromones? And the problem is this: we haven't identified any specific pheromones that go from one person to another. But there is some evidence that pheromones exist. We just don't know what they are. What is the evidence that they exist? Part of the evidence comes from the fact of a thing called menstrual synchrony. I don't know if you guys have heard about that, but often women living together will end up with their periods at the same time, even though when they moved in together, they weren't at the same time. Now, many scientists believe that the reason why menstrual synchrony occurs is because cycles in line with each other so they can all be menstruating at the same time. I don't know. Noticed that happening. That may be due to pheromones, but the reason why it's a problem is because we haven't identified any specifically within humans yet. Would pheromones be endocrine or exocrine? They'd definitely be exocrine, because exocrine means going outside the body. Endocrine means going inside the body. So if it's a hormone, it has to be endocrine. If it's a pheromone, it would be exocrine.
Could you say that? Oh, sorry. Could you say that males tend to notice when a female ovulates in a way to recognizing that they're more attractive, and that could be an indication of pheromones too?
Now, I'm gonna go out on a limb here. It could be due to pheromones. Let's see, we've got a chat here. Which chapters are on exam 2? Well, it says right in module 3, 12, 17A, and 17B. It says it right on the Start Here page, guys. I just went ahead because we're supposed to be lecturing on next week's material now, so that you guys have time to work on review questions before next week's review. But we're playing catch up at this point, and now we're in chapter 12. Okay, so I need to address the question of male attraction. Now, there's two possibilities, and there's a fairly good chance that women who are ovulating are giving off pheromones and guys are picking up on it. And if that's the case, it wouldn't require the woman to be doing anything differently. But there's also some scientists think that when women are ovulating, there's a couple of possible things that they could be doing. They could be behaving differently in a more attractive way, or they could be dressing differently or using different perfumes, or just coming off with more appealing style. I don't even know. But it is, most people believe that when a woman is ovulating, it's fairly obvious to people who are paying attention closely. And that could be due to pheromones that she's just giving off and getting to the males, or something that she's doing subconsciously or consciously to indicate that she's ready to make a baby. And not every woman who's ovulating wants a baby, but your body, when you ovulate, wants you to get pregnant, because that's the biology. Hormones are always made in endocrine glands or individual cells. They're always transported by the blood throughout the body, and they end up at a place called a target tissue receptor. Now, that would be a receptor. We talked about receptors. They're proteins found on the surface of the cells. And if they have the correct shape, the hormones are going to stick to it and cause a chemical reaction inside of the cell. There's two possible ways that can happen. Either the hormone goes inside the cell to cause a reaction, or it sticks to the surface and causes a chain reaction inside without actually going inside. We'll talk about both of those ways that works here in just a moment. It always activates a physiological response. That means something chemically happens inside of the cell when the hormones stick to the receptor on the surface. But here's the thing, not every cell has receptors for every hormone. So hormones are only supposed to stick to the cells that have receptors. When they have receptors, the hormone has an effect. Why is the hormone activated only just before entering the bloodstream? Why not produce it in its active form? The answer is because you don't want the hormone acting on the cell that produced it. You only want it acting on the cell that is the target. So for example, insulin, you guys have all heard of insulin. That's a hormone, and it's produced in an inactive form, and as soon as it leaves the cell, it becomes activated so that it will only affect the cells for which it has a receptor and not the cell that produced it, which is your pancreas.
So if they acted on the cells that produced it, would it just cause it to get denatured?
Not necessarily, but the cell that produced it doesn't want to have that working right in it. It's kind of like people driving the cars around inside a car factory, right? The idea is if you're making something, you send it out, and then when it arrives, it has its effect, right? You don't want it doing that in the cell that made it because it's going to cause problems, a variety of problems. You only want the cells to be affected when a hormone from a different cell sticks to it or comes inside, then it has a response. Does that make sense?
Yeah.
Okay.
Would it
All
be
right,
fair to say that it is to help maintain homeostasis?
Yes, good. That's a good one. I like that. Okay, back to it. I hate to rush you guys, but we have so much to do today and so very little time. I sound like Willy Wonka. All right, if it's a surface, and this is what's showing here, the hormones stick to the outside, it causes one of two things to happen. Generally, it can either open an ion channel or it can activate what is called the second messenger system and a cellular response. This particular figure comes from a different text, so this will not be on the test, but it shows you two ways that peptide hormones act on their target cells by sticking to two receptors and causing a chemical reaction. What did you guys hear me say?
Can cause a chemical reaction.
Cause a chemical reaction inside. So something happens inside the cell. So surface receptors bind the hormone, you see it happening right here, and cause a cascade reaction. Now, what is a cascade reaction? It means one thing happens that causes another thing to happen, and a third, and a fourth, and a fifth. And we've already seen that earlier in the semester, but that's a very common thing to happen inside of our cells, a chain reaction of different reactions leading to an ultimate final product or reaction inside the cell. Okay, this is not going to be on the test, but it kind of shows you how peptide hormones work on the cell. Amine hormones. Now, these are much smaller. They're made up of either 1 to 2 amino acids. That's why they're called amine, because amine is referring to the amino portion of the amino acid. It always contains a benzene ring. If you have had organic chemistry, you know what benzene is. If you haven't, you'll probably end up taking it if you're getting a bachelor's degree in nursing or a bachelor's degree in one of your pre-health classes. You'll learn about what benzene is. It's a really common molecule in organic chemistry. Okay, so what are some examples of amine hormones? These are some ones that you may have heard of. Thyroxine. This is one of your thyroid hormones responsible for metabolism. Epinephrine. You guys have heard of adrenaline, right? Epinephrine and adrenaline are the same thing, fight or flight response. And melatonin, you may have heard of this. Some people take it as a supplement. We're going to be talking about why that's a problem to take melatonin as a supplement. The absence makes you want to wake up.
Oh, dang. So you said we're going to be talking why melatonin is a problem, and then you froze.
Yeah. It's the same reason why taking anabolic steroids that simulates testosterone is a problem. It's called exogenous medication, and it makes the problem worse. It doesn't make it better. And I'll tell you why melatonin is a problem and why I never take it. And it has to do with the physiological response of use it or lose it. But don't let me forget.
Isn't it? Oh, go ahead.
Oh, so I was going to just understand the use it or lose it. So basically, because you're getting supplemented melatonin, if you go off of it, your brain won't make adequate amounts anymore and ruin your circadian rhythm.
Right.
Oh, okay.
But the main issue is if it's coming in from your diet, the pineal gland never needs to make it because you already have enough and it starts to shrink. So when you go off of it, your pineal gland has shrunk and it can't make as much. And now you have the problem worse than you had it before. So then
Does it ever recover?
Yeah, it can recover, but you would have to stay off that hormone for weeks before your pineal gland would recover. I recommend never take anything with melatonin in it because what it does is it tells your body, hey, you don't need to make it. I already got plenty. And we're going to be talking about that reaction, that negative feedback mechanism later on. Okay. So
is this a situation as well where if you continuously take that supplement, your body becomes dependent upon the supplement?
Catherine?
Yes, is that a situation where your body becomes dependent upon the supplement because your body is no longer making its own?
Right. Yeah, that's the problem. And the same thing happens with testosterone like anabolic steroids. The male's testicles shrink because they're no longer needing to make testosterone because they're getting it as a supplement. Okay. So a guy who goes off anabolic steroids is actually going to become more feminine than he would have otherwise been because he's not producing the testosterone anymore because he's taking it exogenously. I don't want to get on a tangent. I got to keep going through the notes, but we are going to talk more about it. All right, steroid hormones are derived from the molecule. Ever studied cellular anatomy, smooth endoplasmic reticulum is called smooth because it doesn't have the studs of ribosomes on the surface, which would be for protein production. They have to be transported by a hydrophilic protein. Now, the question is why? Well, if it's oil-based, it's oil. By the way, all of this is oil-based because cholesterol is oily. The smooth endoplasmic reticulum makes lipid-based molecules, which are oily. Your blood is mostly water. So if you have something oily, it can't move through the water because oil and water don't mix. So the hydrophilic protein means the protein does mix with water. So one end of the protein sticks to the oily steroid hormone and the other end sticks to the water so that you can move this through the watery blood. They're lipophilic means that they can easily diffuse across the plasma membranes. What that means is when they arrive at their target cell, they don't have to necessarily go through a channel. They can go right through the membrane and make it into the cell. If you guys remember when we talked about Fick's law, if a molecule is oily or non-water based, it can go right through the membrane more quickly. And that was one of the things I wanted you to talk about in that essay.
So wait, then why does it have to be transported by a water attracting protein, a hydrophilic?
Okay, because your blood is water, right?
Mm-hmm.
Mostly water. And if you're going to transport something oily through water, that doesn't mix. It won't go. So you have to have a part that sticks to the water so it moves through the watery blood and then can detach and then make it through the membrane. But the idea is if it's oily, you can get through the membrane easily, but it can't go through a watery medium very well. Does that make sense? You have to have a separate protein moving it through a watery solution, but it can go through the membrane on its own because the center of a membrane, a cell membrane, is oily.
But the middle part is not, it's watery. And that's where it would get, the
Right,
middle part of the membrane is watery, and that's where it would need the hydrophilic protein to get through the middle part.
Is watery and your blood is watery, so you have to have proteins carrying those oily molecules.
Yeah,
Almost
I
you
feel
can
so
kill
bad
my
for you. Your internet.
Yeah, it's worse, and it's not even windy here. Usually it's bad when it's windy. It might just be because a lot of people are on this particular lecture.
Yeah, and unfortunately, I didn't even hear the
Oily, so it doesn't need a carrier in there, but in the blood, which is watery, and the cell, which is watery, it's got to have a carrier because it's oily, and oil and water don't mix. That's the best I can do for now. Gotta move on, guys. Okay, so the idea is they enter the target cell and usually to the nucleus. The key here is what is called the genomic effect. That is, it directly affects your DNA. It tells the DNA usually, hey, start making messenger RNA, which is then going to be transported out of the nucleus to a ribosome to make protein. But the idea is that when these steroid hormones arrive, they make their way to the nucleus, they affect your DNA, and they usually stimulate protein synthesis, or they could perhaps stop protein synthesis. But either way, they affect your DNA, and that's why it's called the genomic effect, because they're affecting your genes. Does that make sense, guys?
Would that be the answer that we would use on our review questions for when a molecule has a direct effect on a DNA? What is it?
It's genomic.
Genomic.
Very good.
genomic.
I'm glad you've already made that connection between the review and the lecture. And normally I like to do the lecture first and the review question second, but and really I probably should have done 2 lectures a week during the summer, but I got a lot of stuff going on and you guys have a lot of stuff going on. And I didn't want to ask you guys to come twice a week. Once a week is hard enough. Anyway, we'll figure it out. So
Would you be opposed to recording a lecture for like a chapter throughout the week and then posting it for us to listen to it? So that way, if we do get behind, we can listen to it on our own time?
Without us having to be there.
I generally don't lecture to an empty computer.
Oh.
So what we'll do is I'll just go through as much as I can on Tuesdays. And then if you got to leave you can always come back and watch it, but I'm just going to roll until we get everything done that we got to get done. And then that way you can watch it. And the reason why I don't like lecturing just to an empty computer is because I don't get any interaction with any of the students. And a lot of the learning comes from my interaction with you guys. So yeah, it would probably be better to have it twice a week, but I don't want to just sit down here and lecture into a computer. It feels weird. I've never done it. I probably never will. Some teachers do it. I don't like it. I'm not going to do it, but it's a good idea. I may end up doing it, but probably not. So stick around as much as you can, and find where she left off and continue to take notes. All right, guys, we need to focus on these notes because we're still behind. So what are some examples? Cortisol, which is a stress hormone produced in the kidneys, estrogen and testosterone produced in the gonads. By the way, estrogen is produced in the gonads because the female ovary is a gonad. They're not just male organs. Okay, so this is how it works, guys. This is from your book. It's not my favorite figure, but it works. Okay, so here's your lipid-soluble hormone. How does it work? By the way, what it doesn't show is that the lipid-soluble hormone is being carried by a protein-based carrier molecule. So just note, I'm telling you, as it's going through the blood, it's going to be being carried by a protein transport molecule. And then it leaves the capillary and a receptor. So the receptor takes it from the, by the way, the receptor catches it right as it passes through the membrane, and then it transports it all the way into the nucleus, and it finds its way to the DNA. What it does is it activates the DNA and causes a process called transcription, which is the copying of a section of DNA into a piece of messenger RNA. Notice this DNA is double-stranded. The messenger RNA is single-stranded. By the way, RNA is what it's made out of. The M is its job, which is messenger. A messenger takes information from one place to the other, in this case from the nucleus out to the ribosomes. Each of these orange things is a ribosome. So this messenger RNA now finds its way out through a nuclear pore, finds a ribosome, and this picture shows the messenger RNA feeding through several different ribosomes, and the protein comes out is based on what it said in the DNA. So now we have the protein that was made as a result of this hormone coming in, telling the nucleus to make a copy. This copy then ends up being the code for the production of the protein. Does that make sense to you guys?
Yes.
Yeah.
Okay, so this, so I'm glad I was able to talk to you about it. And there's more information about it in the text if you're interested.
Oh, what did you say the lipid soluble hormone is being carried by?
Go
Is it
ahead.
a hormone? Did you say the lipid soluble hormone is being carried by a protein carrier in the blood?
Right,
that right?
and I mentioned that in the previous slide.
Yeah.
That these lipid soluble hormones have a protein carrier. It just doesn't show it in this particular picture. So just imagine there's a protein. You guys see this blue thing right here? They should have shown it here as well.
Does
So
it get released once it gets out of the blood?
Right. So what it does is that what happens is the protein carrier releases the hormone, it passes through the capillary. And by the way, the side of the cell membrane and the blood vessel are going to be right next to each other. So it just goes from through the blood vessel, through the membrane, and then it picks up another receptor, another protein carrier when it's in the cytoplasm. Because remember, the cytoplasm is water, the blood is water. So if it's in a watery solution, it's going to have one of these protein carriers. And that's what you're going to want to include if this ends up as an essay on the test. And by the way, at the end of today, I'll be actually showing you the essays that are actually on the test, so you can know exactly which ones you have to study and get ready for your test on Friday. True or false, everybody? The best time to take the test this week is on Sunday evening.
False.
Why?
Because rest on Sundays, and you want to prevent any possible errors that might have to.
Do you know how many
delay you.
people took test 1 on Sunday, this last Sunday?
I was one of them.
I did, but that's what worked best for me
60%
this
of you
week.
guys didn't listen.
Right.
Now, I will say, I did talk to somebody who was a student recently, and they said sometimes Sunday is the only day they get to do
Mhm.
stuff like that. But I mean, unless that's the case, really, you don't want to wait until Sunday. And Sunday should only be if there's stuff that you couldn't get done during the week. I mean, you know, it doesn't matter to me. You guys, you take the test, you don't. But I'm wanting you to be successful, and the most successful students don't wait until the last minute to do things. That's what that whole week one, discussion board is about, right? Don't procrastinate. But some people have really, really busy lives. I get it. If that's all you got, and that's all you got, I won't give you a hard time, but you're probably going to hear from me Saturday afternoon if you haven't done the test by Saturday afternoon. Try to get it done no later than Saturday night. Take Sunday off if you can. All right, got to move on, guys. All right, we're going to be talking about an endocrine reflex pathway, which is part of the reflex system. We're going to be talking about reflexes more later, but the endocrine reflex pathway means you get stimulated. It takes you out of homeostasis. And to get back into homeostasis, you have to do something. That means the endocrine system has to respond, getting you back to normal. So the steps are stimulus, and that is something gets out of normal, away from homeostasis or normalcy. What that does is it sends an afferent signal towards the central nervous system. And what does that mean? It means that a signal from the body towards your brain or spinal cord. There's integration in the brain or in the spinal cord. Usually it's in the brain. The efferent signal is the hormone is released. That means something is being done to fix the problem. The physiological action happens, basically trying to get you back to normal. And negative feedback happens. And this is what negative feedback is. It means that when you're out of normal, you're doing something to fix the problem is gone. And so negative feedback means that you get the signal that everything's a-okay. There's no more signal saying you have to do something. And as soon as the signal telling you to do something disappears, you stop doing the thing and you're back to homeostasis. So negative feedback means the hormone did the thing that got you back to normal, and now you don't need to do anything anymore. That's why it's called negative feedback, because it goes back to the beginning saying, we're good. You don't need to do anything anymore because you fixed the issue and got yourself back to homeostasis.
Can I get clarification on number 3? Was that subconsciously? Is that the answer to that question?
It would be subconsciously, right? So this is incorrect. If it's a reflex, it's not conscious, especially with endocrine. You don't know it's happening. You never know it's happening. It's all happening behind the scenes in your subconscious brainstem, hypothalamus, pituitary. We'll be talking more about that.
So you could say all reflexes are subconscious then?
Yes, they are. Every reflex, and that's why it's called a reflex. Because if I decide to move my finger up and down, that's not a reflex. But if somebody starts to push a ball toward automatically. And that's true for every endocrine reflex pathways. It's unconscious because you're not aware of it. Did I cut out?
Yeah, you just said that's true because of every endocrine pathway.
Every endocrine reflex is subconscious because you don't know it's happening. It just happens behind the scenes. Okay. Now, this figure, even though it's not in your book, may be on the test, and I'll let you know later today. But this is a really good figure that shows you how endocrine reflexes work. And this is from a different text, but it's a really good one, and you didn't have anything like it in your current text. So what I'm going to do is I'm going to be talking about how this reflex works. I want you to take notes on it, and then I'll let you know whether or not this is going to be an essay on this week's test. Okay, so one of the things that most people believe is that insulin only gets released when you eat a lot of sugar. That's what many students believe. It turns out that that's not true. There's 2 different pathways that stimulate your pancreas to make insulin. And what we're going to do is we're going to talk about the traditional pathway, and then we're going to talk about the pathway that some students don't know about. So when you eat a meal, if it contains a lot of sugar, glucose, what happens is your blood glucose rises. So that causes a rise in blood glucose. The blood glucose makes its way to the pancreas. The pancreas responds to the increase in blood glucose by producing insulin. The insulin leaves the pancreas and goes into the blood. Then it sticks to the surface of certain target tissues, and what it does is it causes these target tissues to pull sugar out of the blood and store it in those cells. When you pull sugar out of the blood, it causes your blood glucose to drop. The negative feedback, which was stimulating it, which means, hey, we're not out of normal anymore, goes back and says, you don't need, it kind of loops all the way back to the pancreas and says, we're good, we don't need any more insulin. When your blood sugar gets back to normal, which is right here, the pancreas doesn't get stimulated anymore, it stops making insulin and everything shuts down. What caused it to go do its thing was the rise in blood glucose. Blood glucose go back to down to normal. All right. The other way that you can release insulin is when you eat a meal that causes your stomach to stretch out, even if there's no sugar at all. Let's just say you had a big steak, not a bit of sugar in there. It still stretches out your stomach, which sends a signal through the afferent neuron to your brain or your spinal cord, usually your brain. Then the signal leaves the brain and goes to the pancreas and says, hey, this person has a full stomach. So we're betting, or we're a betting brain, that there's going to be some sugar in there, even if there isn't. It doesn't, your brain doesn't know. And it says, so we're going to go ahead and produce insulin just in case there's going to be some sugar coming out. And so what that does, and one of the things you may have noticed is if you eat a large meal that doesn't contain a lot of sugar, sometimes you can get very drowsy. Often that happens because the insulin is produced, it goes to the target tissues, causes your blood sugar to actually drop below normal, and you end up with low blood sugar. That happens because the body assumes that when you have a full stomach, some of that is going to be sugar. So the idea is, if I ask you about this, there's two pathways for the release of insulin, either directly as a result of blood glucose or as a result of a full stomach, whatever. Both stimulate the pancreas to produce insulin. The insulin goes to the target tissues, causes blood glucose to drop, and this stimulus only diminishes when you finish digesting your food. Okay, so there's two possible ways to stimulate the pancreas to produce insulin. Now, is it true that many large meals also have lots of sugar in them?
No.
Like, so you're saying that there's not sugar in large meals, like
Yes.
the meals that you would get at a fast food place, like a Happy Meal.
Oh, well,
Starches
that, yeah,
convert
but I
to
thought you
sugar.
meant a
Combo
balanced
meal.
meal.
Usually it's in the French fries, in the bun, and in the soda, especially in the soda, right?
Starches.
So, like sugar. So not only are you stretching your stomach out with this big meal, but you're also adding a bunch of blood sugar. So what it's doing is it's double stimulating your pancreas, which causes even more insulin to be released. Now, the reason why this is significant is you guys have all heard the story of the boy who cried wolf, right? If you cry wolf too much, people stop believing you. Now, it turns out that if the pancreas is producing too much insulin all the time, the target tissues What was the last thing you heard me say?
You were good. You didn't freeze that time.
Ah, okay.
Mine did actually. It said it just targets tissues and then you froze.
Okay.
But
So if there's too much insulin hitting the target tissues start pulling receptors off of the surface because they become desensitized. There's so much insulin that they start pulling receptors off because there's just too much stimulus happening. So what happens then, it takes even more insulin to make the sugar come in, and that's called type 2 diabetes. So type 2 diabetes often comes from the fact that you are putting too much insulin in because you put too much sugar and food. And what it happens is the target tissues don't have the receptors as much as they used to, and they can't then absorb the sugar, and the cells start to die because without sugar, the cells can't live, and you can't get sugar into your cells without the insulin helping with facilitated diffusion, something we talked about earlier in the semester. So how do you cure type 2 diabetes? Stop eating so many large meals and so many meals with sugar in it. And my father actually cured himself of type 2 diabetes by having smaller meals with less sugar and starch in it. Now, would
So-
that happen for everybody? I don't know.
So does this work this in the same way that like addiction starts to accrue by like you have an influx of the substance and the
Right.
more that substance, the receptors get pulled away so you don't get so overwhelmed?
Like cocaine, right? Cocaine stimulates a lot of pleasure the first few times, right? And then the body
Yep.
starts removing receptors for it. So it takes more and more and more of it to get the same response. But you can't keep producing more and more insulin because the pancreas can only produce so much. So it turns out that even normal amount of insulin is not going to have the effect of getting sugar into the cells. And yeah, it's just basically a version of addiction. You're putting too much insulin in because you're putting too much food and sugar in, and that's overwhelming the system and making it lose its sensitivity in the same way drugs do. So if you want to lose type 2 diabetes, one of the ways you can do it is by having smaller, more frequent, less sugar rich meals. So if you're getting a super size Happy Meal with a sugary soda, you're setting yourself up for diabetes. Or
How
just
could
eating worse.
exercise?
Exercise helps because it causes your blood sugar to drop, but you can
It
only
doesn't
get
hurt.
away with eating really, really sugary, starchy food if you're exercising a lot. Most people don't. They eat a lot of sugary, starchy food, even though they're couch potatoes, because when they were younger, they were much more active and they got in the habit of eating the certain foods that they eat, or their family eats a certain food that they eat, and you get used to having a certain thing, and you don't need all those calories, especially those sugar-based calories. Our brain likes sugar because it's a quick, easy source of energy if we're very active. But unfortunately, we have the same hunger and desire for sugar, even if we're not active, because our body assumes that even though we're not active now, pretty soon we will be, and we want to store some extra energy in the form of fat or glycogen in our livers and muscles. Does that make sense? There's so much to tell you guys about so little time. If you guys took this in an 18-week class on campus, I would be able to go into all this in great detail and we'd have some great hands-on labs, but you guys didn't do that. All right. It's just not the situation we're in right now. All right. That's all I have time for on this, but do your best with it. And so that was what, 12A?
This is 17A.
Are we
That was-
able to touch on any of the questions, the review questions, or no?
Well, do we need to do one more lecture?
I
Two more.
What did we just cover? 17A?
Technically 3.
Yeah, that was 17.
Okay, so
But
look,
we
this
have
is what we'll do.
17B still.
Okay, I'll do 17B. It's fairly short, and then we'll do review. And anybody who wants to stick around, then we'll do 12. Okay. All right, I know we're way behind. It sucks, but I will do my very best to get caught up next week so that we're completely and totally caught up. I think one of our upcoming weeks, we don't have as many chapters and we don't have as many other things to do, so we should be able to get completely caught up. Let's see, where are we? Zoom, share screen, 17B. Okay, here we go, 17B, and then we'll do a review for the exam as soon as we're done with this. Okay, so we're continuing our discussion about hormones, and the human nervous system produces several major groups of hormones. The first is called catecholamines, released by neurons in the adrenal glands, which are the suprarenal glands sitting right on top of your kidneys. Hypothalamic neurohormones, basically those are hormones produced by the hypothalamus, and those control your thalamus. Your hypothalamus is right here. Do they show that? They don't show the hypothalamus. It's kind of unfortunate. The hypothalamus is right here.
It is showed in other pictures that we have in our printout.
Okay, good. So the hypothalamus is right here. Right below it is the pituitary, and this shows you all of the major endocrine glands. The only thing it doesn't show is the hypothalamus right here. Really should, but the pictures in these books, this book is not great. The pictures are not great because it's a free book. And anyway, I've been pressured, by the way, to adopt free books, and the free books don't have quite as good of figures as the regular books from the publisher. Anyway, okay, so here's a pretty good picture. You guys see this blue area here? This is called your hypothalamus. So the way that it works is these, and by the way, we're talking in this particular picture about the posterior pituitary. What happens is the hormones are produced here in the hypothalamus, and then they get transported. You guys see they're transported down here, and they get released at the posterior pituitary. The point I want to make about the posterior pituitary is it does not make any hormones of its own. What it is, it's just a site for the release of hypothalamic neurohormones. So the hypothalamus is making them, transporting it through what is called the infundibulum, or the hypothalamo-hypophyseal tract or portal system. And then it's getting dumped here when a signal from the hypothalamus comes down. So this is just a point of release for hypothalamic neurohormones. The posterior pituitary doesn't produce any of its own hormones. That's the point of this picture. Okay, this is only the place where they're released into the blood. It's weird. Why not just release it up here? I don't know. It releases it here. So this is the posterior pituitary. What we're going to see is that the anterior and posterior pituitary have very different jobs and have a very different release mechanism. Where in the neuron are the hormones of the hypothalamus produced? It's got to be at the cell body, because that's where the nucleus is, and that's where all, if you want to do the construction of molecules, you have to do it near the nucleus, because the nucleus is responsible for transcription and translation. Does the posterior pituitary actually produce any neurohormones? What did I just say? Yes or no?
No.
No, it's only a site for the release of the hypothalamus hormones. Okay, the pituitary, talking about it in general. It's a lima bean-sized gland below the hypothalamus. It's composed of two parts, the anterior and posterior. As I told you, the posterior doesn't make any of its own hormones. The posterior pituitary is just for storage and release site for two kinds of hormones. The first is called vasopressin, which regulates how much water you maintain in your urine, and oxytocin, which controls milk delivery and contractions during labor in women. So that's the main job of oxytocin. You can also use oxytocin to induce labor if a woman needs to be induced. Anterior pituitary gland. This is very different because it is going to be actually producing hormones. But here's what's weird. It produces its own hormones, but it has to be told by hormones from the hypothalamus to release the ones that it is making. So it's basically waiting for a signal to release the hormones that it has made. So it secretes six basic types of hormones. Prolactin, which basically helps women produce milk, not release it, but just make it. Thyrotropin, or thyroid stimulating hormone. Here's something you guys should know. When you see this suffix tropin or tropic, it means a hormone that is destined for another endocrine gland. In this case, it's thyroid stimulating hormone. So it means that this thyrotropin is going to stimulate the thyroid to produce metabolic hormones. Adrenocorticotropic hormone, or adrenocorticotropin. Adrenocorticotropic hormone. So what it's saying is, there's your adrenal gland sitting on top of your kidneys. The cortex is the outer part. Corticotropin means it's going to be acting on it. So what it means, it's a hormone that stimulates the adrenal cortex to release its hormones. Human growth hormone, you guys have heard of that. One of the main jobs of human growth hormone is to stimulate muscle cells to expand. That's why athletes take it. It's also present in high levels all the way from the time you're born to the time you mature. That's why you get taller and stronger as you're maturing. Follicle stimulating hormone works in both males and females. It does affect the reproductive cells. Luteinizing hormone, primarily found in females, affects the corpus luteum, which is part of their reproductive system that we're going to be talking about towards the end of the semester. Most anterior pituitary secretions are tropic, which means that they go to another endocrine gland and tell it to release its hormones. So here's the logic, guys. Your hypothalamus, which is up here, tells your pituitary, which is right here, to release hormones. It releases the hormones which go through your whole body and hit other endocrine glands. They release their hormones. Those hormones affect your cells. Now you have your effect. So hypothalamus affects pituitary, pituitary affects endocrine glands affect your body cells. So, quick question. True or false, your brain is responsible for most of the biochemistry in your body, indirectly. What do you think?
True.
True. Yeah, because your hypothalamus is actually a part of your brain. It tells your pituitary what to do. Your pituitary tells your endocrine system what to do. Your endocrine system tells your body what to do, which means that your brain is in control of your body. But is it conscious, do you think? Can you control the release of these by thinking about it?
No.
No. So it's your brain does control it, but your conscious mind does not. So there you go.
If it did,
Okay.
I'd be taller.
What's that?
If it did, I'd be taller.
Yeah, everybody would have all kinds of changes to their body if you could make, you'd probably book, but it does help you understand how this works. Okay, this figure is not in your text, but it does help you understand some of the hormones and how they work. Everything in pink is being produced by the hypothalamus. Its target is the anterior pituitary. And you guys notice the hormones are being produced in the hypothalamus, and they go through a thing called the hypothalamo-hypophyseal portal system. What is the portal system? Normally, blood goes from your heart to a capillary bed, and then from the capillary bed back to your heart. In a portal system, it goes from a capillary bed through some vessels to another capillary bed. That's why it's called portal, because it's porting blood from one capillary bed to another. In this case, it's a capillary bed in your hypothalamus to a capillary bed in your anterior pituitary. So when these hormones come to the anterior pituitary produces these. When these get stimulated and released, these go to all of these other cells, the thyroid, the adrenal gland, the liver, and the gonads. Then when these get stimulated by your anterior pituitary hormones, they release these functional hormones, thyroid hormones, cortisol, insulin-like growth factors I'll be talking about in a few minutes, androgens, primarily in males, and estrogens, progesterone, primarily in females. And then you get, by the way, the actual effect, which is what these are doing. So we have the hypothalamic hormones affecting the anterior pituitary. Anterior pituitary affects these endocrine glands. Endocrine glands affect many tissues. You guys see how that works? And it's actually showing it to you at each level. Now this picture will not be on the test. Okay, now we're going to talk about multiple stimuli for the production of
Did
blood.
you say anything before or after you had said this picture will not be on the test? Because you froze.
I don't believe it will, but it does help you understand the sequence of events between the hypothalamus, the anterior pituitary, and the endocrine glands. I don't believe this is on the test. I went through and I think I pulled it out because it's not in your book. What I'm trying to do is not put any figures on your test that are not in your book, because then you won't be able to look at them in your book to get some help with them. Okay. Generally, that's what I've been doing. Trying to make sure that if it is on the test, it's also in your text.
Muchly
I may be-
appreciated.
Yeah, you're welcome. I'm trying to make this as fair as possible. I know it seems like a huge amount of information, a short amount of time, but I'm trying to make it as fair as I can. All right, we got to go back to this. Blood sugar balance. We talked about this when we were talking about the production of insulin. The stimuli for the production of insulin by the pancreas. This is talking about the figure from the previous lecture, which was 17A. Stimulation of stretch receptors in the stomach after a meal. I mentioned that. Or increase in blood glucose levels, which was the other side path. The reflex, though, is the production of insulin. What the insulin is doing is to try to get your blood sugar down. This lowers blood glucose, reduces the stimuli, which was the original increase in blood glucose levels. That causes the pancreas to produce less insulin, and you're back to homeostasis. So that figure, by the way, was, this should be out of here. The figure was the one that we already had. Okay, multiple hormones can target a single cell or tissue. So growth hormone can do it.
Did you skip the slide impacts of multiple hormones on targets?
This one?
Yes.
Okay, I'm sorry. I don't know how these got out of order, but is this the next thing in your notes? Is this the next part of your notes?
Yes.
Okay, so I'm gonna have to back up, I guess. I thought I had these in order, but switching from one text to another text can be challenging. All right, so the term is synergism. Multiple stimuli are more than additive. Cortisol adds plus 5 milligrams per 100 milliliters of blood of sugar to your blood. Glucagon adds 10. Epinephrine adds 20. And if this were cumulative, or excuse me, if this were additive, it'd be 35. But the synergistic effect of all of them together is plus 140 milligrams of sugar per milliliters of blood. So that's why it's called synergistic. They working together amplify each other rather than just adding together. Another term is called antagonism. That means you'd never have them be at the same time. So insulin is when you have too high of blood sugar and you're trying to get it down. Glucagon is when your blood sugar is low and when you're trying to raise it. So glucagon is produced between meals when you're hungry. Insulin is produced after you've eaten to get your blood sugar back to normal. Permissiveness is that the main hormone needs a second or third hormone to show its full expression. And reproductive system is the best example of that. That even though you do have some reproductive hormones, having other hormones along with it allows that reproductive hormone to take effect. Okay, what do you guys have next in your notes? Oh, by the way, this is the figure that shows the synergism. So you got synergism there. This is the figure showing synergism. What's the next thing you have in your notes?
Pathologies over or under production.
Okay, so I guess there were several slides in my PowerPoint that didn't end up in your notes. So if it does end up as a review question, we'll have to go over it. Okay, so pathologies over or under production. So there's no bad hormones, just too much or too little. There's no such thing as a bad hormone. Now, I made an allusion to this. I referenced it earlier, that if you take a hormone as a supplement or an injection, that's called exogenous, because exo means from the outside. You can inject or ingest it. It replaces and exceeds the normal amount that your body needs. The problem with that is it basically stimulates negative feedback and says to that gland, hey, we don't need you anymore. We're getting plenty on our own. You don't need to make any, and it makes it shrink. So it causes atrophy. Atrophy means shrinking. So that's if it doesn't get stimulated, it shrinks. So when you take melatonin, it makes your pineal gland shrink. And so if you get off melatonin, you're going to have even more difficulty sleeping. And that's exactly what those companies want. Because if you're taking it constantly and you know you can't get off it, then they've got you hooked. And that's true for a lot of prescription medications as well. They want you to become addicted to it because that's how they make their money. Sounds like a conspiracy theory, but it happens to be true. All right.
As they say in the medicine world, there's no money in giving a cure.
Right. They don't try to cure you. They just want to keep you hooked. That's why I don't take any prescription meds. I try to figure out something else to do besides getting on medicine, because once you're on it, you're kind of stuck on it because it doesn't make you any better. It just gets rid of the symptoms. Generally, normally. All right, a good example, testosterone, the effects would be shrinking the testes. Why is that the case? Because normally your brain stimulates your testicles to produce testosterone to maintain levels. But if the levels are already there, there's no stimulus for the testicles to produce it, so they shrink. So bodybuilders who take a lot of anabolic steroids, which mimic testosterone, have their testicles shrink. Question, do bodybuilders really care about the size of their testicles, or is it the size of their muscles that matter?
As the muscles.
Most guys kind of do care about the size of their testicles, or at least the ones I know about, but bodybuilders are very focused on their muscles. All right, hypersecretion. Now you might guess this guy with goiter is hypersecretion, but it's not. I'll tell you why. Too much hormones produced. The best example is Andre the Giant. If, by the way, if you guys haven't seen the movie knowledge is to watch Princess Bride, because it's such a great movie, and he's in it. He's an actor. And
Inconceivable.
It's a great movie. What'd you guys hear me say last?
Watch Princess Bride.
Yeah,
It's a great movie, is what you said.
He's in it. He's really funny, and you'll see how big he is. Now, why did he get so big? Because his body produced too much growth hormone, both before and after he stopped growing. So some people who produce too much growth hormone right up to maturity, they're tall and slim, but he continued to produce it after he stopped growing taller, and that's why he ended up so massive. He used to drink like 5 bottles of wine and like 20 drinks in one night because he was so big. Anyway, Okay, so it can be caused by tumors or cancer. We don't know what caused Andre to produce so much human growth hormone, but some people end up, by the way, if you look at how thick his fingers are, his brow ridges on his forehead, that is
that
what is
like
called acromegaly.
acromegaly? Oh, okay.
Acromegaly is what happens if you have a growth hormone after you start getting taller. The symptoms of it are thickened fingers, thickened tongue, and that's why he spoke with an unusual accent, not just Belgian, but he had a thickened tongue, thickened brow ridges. That's what happens. Too much growth hormone after your bone plates fuse and you can't get any taller. So gigantism and acromegaly, he had both. So not only was he tall, by the way, the guy he's standing next to is 6'6", which is usually pretty tall, right? Andre was over 7 feet tall, and he weighed 550 pounds with very little body fat. So he was a really huge guy. So he had both gigantism and acromegaly. Acromegaly happens if it happens after you stop growing taller. Gigantism is if you just get taller before you stop getting taller, but he had both. Okay, so the opposite is hyposecretion. And this guy with goiter actually suffers from hyposecretion. So how does this work? Well, let me explain it. If you remember, I talked earlier about how your brain sends a signal to your thyroid gland to produce thyroid hormones, right? Well, what if the thyroid can't respond and produce the hormones that is being told to be produced, right? It will just keep getting stimulated because the body's saying, hello, hello, is anybody in there? I'm trying to get you to make this thyroid hormone. So it keeps sending the signal down. But because the thyroid can't respond, it keeps getting stimulated and it keeps enlarging. Does that make sense?
Yeah.
So if it were able to respond, the negative feedback would shut off the brain and say, hey, look, I took care of what you asked me to do. So stop telling me to do it. But since it can't respond because of a lack of iodine potentially, it keeps getting stimulated because the brain doesn't understand why it can't make the thyroid hormone. And because it keeps getting stimulated over and over, it enlarges in the same way. If you stimulate a muscle over and over, it'll enlarge. So this goiter is due to the fact that this man wasn't able to produce thyroid hormone, and that caused the brain to continue to stimulate it. And it just kept getting bigger and bigger. Because
So with this, could we compare this to like the freezer making ice, removing the arm that senses
right.
that there's
It would just start shooting ice out. It would blast open your freezer door and it would make way too much ice because, very good analogy by the way, because the negative feedback, hey, that we've got enough, we've done what you've asked, never gets there. So it just keeps sending the signal to make more ice. And you would end up with way too much ice and it would go all over your floor. Does that make sense? So this is him not being able to respond with negative feedback to stimulus from his brain. So it keeps stimulating and the stimulus makes it expand in the same way a muscle. Diabetes is another one. This would be type 1 diabetes, and that is if your the signal to go to the pancreas gets there, but the pancreas can't respond by producing insulin. So here's the deal, you guys. Type 2 diabetes generally is caused by too much sugar, too much insulin, downregulation, or insensitivity. But diabetes type 1 is that your pancreas just doesn't make enough, not due to any problem of what you've done to your diet. It's just can't, for whatever reason, make enough insulin. So type 1, you don't make enough. Type 2, you make too much, but then your cells become desensitized. All right, pathology. So I mentioned this. Downregulation is what happens to the cells of your bodies when you're producing too much insulin. And the term for that is hyperinsulinemia. So hyper means more, insulin, and emia is the problem. So too much insulin, downregulation, too much insulin all the time, usually due to diet. The target cells remove their receptors, as I mentioned earlier. There's not enough receptors to move the sugar out of the blood because the body has pulled the receptors off due to too much insulin. So it's like an addiction.
Can those receptors generate?
Yeah. So what do you think you would need to do to get those receptors back onto the surface of your cells?
diet and exercise?
Well,
Yeah,
stop
primarily not eat so much sugar and large meals. And yeah, exercise would help. So you can actually potentially cure yourself of diabetes just by changing your diet and exercise. Question, do Americans like changing their exercise levels, or their diet.
They don't
No,
like changing
Americans are
anything.
lazy.
So here's the deal, you guys. Many of you guys are going to end up working at hospitals or clinics, right? Do you know what half or more of all your patients will suffer from that has caused them to end up at the hospital or the clinic?
Diabetes and addiction.
Diabetes. Right, because they eat too much food, too little exercise. Question, can you eat a ton of food and be okay and never get overweight and never get diabetes?
What if you exercise a lot?
Right. You have to be really, really active. And if you're not really active, you should cut back on your food. The problem is the brain doesn't believe you, and it still wants you to eat just as much as if you were active. So that's a flaw in our brains.
I remember when I was on team sports and we did water polo, we had to train for like 5 hours a day, and I had to eat 6,000 calories just to keep up with it. And it was really weird because after the season stopped, your appetite was still as if you wanted to eat 6,000 calories to accommodate the activity level that you weren't doing anymore. So a lot of athletes would gain weight.
Right? Yeah, you can gain weight because you get in the habit of eating certain foods. When I was a teenager, I was super active and I got habits of eating. And only lately have I changed my habits. I was up to about 280 and I started eating less and I've dropped about 50 pounds. But yeah, it's really hard. Your brain does not want you to eat less because we still have that lizard brain that says, hey, you might run out of food next week. And if you don't have a few extra pounds packed on, you're going to die of starvation. The problem is we never run out of food. I know that doesn't sound like much of a problem, but it is for obesity. Solution for the problem, true or false? Medical, like pharmaceutical meds is the best solution for the problem. True or false?
False.
False. What's the best solution for the problem of hyperinsulinemia and downregulation?
Diet and
Free
exercise.
sugar, diet and exercise.
Right. Primarily diet, which Americans don't want to do. All right, pineal gland. So we were talking about melatonin, right? Melatonin is produced by the pineal gland in the brain, and I showed it to you earlier in this presentation. It influences body clocks, sleep and wake cycles. Basically, the production of it makes you sleepy. It's usually secreted when eyes perceive darkness. Why is it that people don't make enough melatonin naturally these days? What do we do that makes it hard for our brain to make melatonin?
Exposed to blue light and the light.
Overstimulation.
Well, any light. Blue light is the worst. Blue light is the worst, but any light. So if you want to get sleepy, what should you do?
Make a dark room and make it cold.
Cold and dark, right? But the problem is if people are in the habit of staying up till midnight, then they just lay there and stare at the ceiling until midnight, right? But if you get in the habit of turning off most of the lights and making it as dark as possible, your brain will start producing melatonin. It'll make you sleepy and you're good to go. So increase in melatonin levels normally causes drowsiness, the absence of other chemicals or problems or habits. Insomniacs may take meditation containing melatonin, but remember what we said the problem is? It actually makes the pineal gland shrink because it tells the pineal gland that it's not needed anymore. Here's another thing that, go ahead. Question.
I was saying, does calcification of the pineal gland also cause like a compromising your melatonin production?
It does. That's fairly rare, but it can happen to some people. But did that happen for you?
No, I just, saw a podcast one time and they talked about what causes, if fluoride really actually causes calcification or not. And they ruled out that fluoride actually doesn't. And it's just,
I don't think it does. I don't think it does. It's a conspiracy theory. Some people really hate fluoridation of water, and there's really no good information that says fluoridation of water is harmful. But, people have conspiracy theories about everything. JFK, the moon landing and a round Earth. We still argue about whether or not our Earth is round or flat. It's just, as a science, whether the Earth is round or flat. Social media is not all good. As a matter of fact, it's often very bad. All right, we got to move on. Melatonin also has an antioxidant effect. That's one of your review questions. Melatonin has
it does have an antioxidant effect.
Right. An antioxidant effect is similar to vitamin C. What does antioxidant effect mean? Well, we have to take oxygen inside of our cells because we need to make ATP, right? But the problem is oxygen creates a chemical called free radicals. The free radicals destroy elements of your cell membrane. And so by having vitamin C, it captures and destroys those free radicals, reducing the damage in your cells. So melatonin helps reduce damage in your body cells in the same way that vitamin C does.
And is an antioxidant just indicative of having an extra electron, and the antioxidant has an extra like proton to bind with that extra electron?
It's not usually that simple. It's usually a more complicated chemical reaction where the free radical, it's not just the electron, the free radical is a chemical that runs around and damages cell membrane proteins. So the antioxidant doesn't just provide a source for the electron, it actually combines with the free radical and prevents it from causing damage to your cells. And more than that, I'd just be kind of BSing. Okay.
Thank you.
Normally, nighttime is when when it's dark is when you produce melatonin, and this is what it is. By the way, this is again a derivation of benzene. So melatonin is derived from the amino acid tryptophan. So interestingly, you guys heard of how tryptophan from turkey makes you sleepy?
But is there really enough tryptophan in turkey to actually make you sleepy?
Well, it's a combination effect. Most of the time you get sleepy just because you have a full stomach and your blood sugar levels drop, but turkey does contain tryptophan. And so what happens is that tryptophan gets converted quickly from tryptophan into melatonin, and that's also what makes you sleepy. So when you have that's why you get sleepy from eating a lot of turkey. So there you go. All right. So what we're going to do now, you guys have been super patient. What we're going to do now is we're going to do a review for the exam this week. I'm going to show you the essays for the exam. And then whatever chapter we haven't covered, we're going to go back and do that, which would be 12, right?
Yeah.
Yes, but 12 is on the exam, isn't it?
Yeah.
It is. So, I mean, it's up to you guys. If you guys want me to do 12 and then review, we can do that. If you guys want to do the review and then 12, we can do that as well. I don't know if it matters that much. We'll get to it one way or the other, but I want to take care of the people who are going to have to take off quickly and who really want that review.
Can we at least just go over all the essay questions so we just know, and then
Yes.
do 12?
Okay, that is a good point. So let's go ahead and jump to the essay questions. I like that. So let me go ahead and find the test. We'll look at the essay questions. Then what I'll do is I'll go to 12 and focus on the essays that will be on. Okay, so let me find your test. Quizzes. So this is exam 2. My computer is kind of stuck. Give me just a moment. There we go. Okay, exam 2. I'm going to preview it. I love living out in the country. Don't you guys love living out in the country?
If it looks like the background that you have behind you, then yes.
Well, it's not. I'm out in the middle of the desert. I wish I lived in a rainforest. Okay, so let's go ahead and I'm going to share with you the exam. Exam 2. Okay, so what I'm going to do, because we're right here and this is one of the essays and it is from chapter 12, I'm going to go ahead and lecture on it right now. So if you guys want to get out chapter 12 and find this figure, I'm going to be lecturing on it right now because there it is right on the test. Okay.
Our action potential stages.
This is the action potential, right? This is how the action potential works in a axon, or really it really works in the entire cell, but it's primarily happening in the axon. But anyway, let me get to the things that I want you to tell me about for the test. Okay. Resting membrane potential is a negative 70 millivolts for a variety of reasons, primarily due to the fact that the membrane is leaky for potassium. It's not that important that you tell me why it's at negative 70, but the book does talk about why resting membrane potential is negative 70 millivolts, and MV is millivolts. Okay, so here we are at rest. So this is a neuron just basically sitting there doing nothing. Then what happens is the stimulus is applied, and you start moving the neuron out of its norm. What happens is sodium and potassium, primarily sodium gates, start to open, okay? And so what's going to happen when you stimulate that, jab that neuron with something, either acetylcholine or some kind of physical stimulus or heat or whatever, as sodium starts to rush in, you're going to get to a thing called threshold. Now, this whole section here is what is called a graded potential. You guys want to write this down. The graded potential is happening in the cell body. And it's basically a rise in the membrane potential due to the influx of sodium into the cell body. Okay, then when it crosses the threshold, this is like base, and the analogy I want to give to you on this is how a toilet flushes. You guys all have, I have different kind of toilets in my house. I have the push button ones on top, but most of the toilets have a little handle on the side that if you push on them, they flush, right? You guys
Yeah.
with me on that? Okay.
Yeah.
What happens if you go to your toilet and just very lightly tap on the handle? Is anything going to happen?
Not really, hardly anything.
So imagine if
Water
you
might
went
start
to your
leaking,
toilet.
but that's it.
What's that?
The water might start leaking out a little bit into the bowl, but that's it.
No, I'm just saying if you just touch it really lightly, like if I were just to touch someone very lightly on the forehead, nothing's
Yeah,
going to happen.
nothing.
Nothing's going to happen. But if I start pressing harder and harder and harder on the handle, eventually you're going to get to a pressure that's going to cause the little flapper to come up, right?
Right.
Flapper is going to pop up and you're going to have a flush, right? And once that happens, you can't stop it. So the idea is the graded potential is how much pressure you're putting on the,
It's okay. It didn't freeze that much.
okay, good. So the graded potential rising between here and here is kind of like you putting slightly more pressure on the toilet. When you get to threshold, that's just enough force so that the flapper comes up and the water starts to rush. So the idea is once you get to that threshold, that amount of force necessary to cause the flush to happen, it's going to go finish the flush no matter what you do. You can't stop it without pulling the lid off and smashing your hand on it or whatever.
So in your example, is the amount of sodium being represented by the amount of pressure, the amount of sodium coming
Right.
in?
So
Okay.
so the amount of sodium rushing in is the amount of pressure you're putting on the handle. It's going higher. If you were like really trying to just get the exact amount of pressure on there so it would just barely flush, you put a little bit more pressure, and a little bit more, and eventually you get to the amount of pressure needed to make the flapper pop. And that's right where threshold is. So as the sodium is leaking into the cell, at a certain threshold, it causes a chain reaction. And the chain reaction then spreads down the axon and it can't be stopped. And it always goes from the cell body down the axon. So at this point, threshold has been reached, and now the chain reaction happens and a whole bunch of sodium is rushing in. So it doesn't show it here, but it'll tell you about it in the book. This big rise is sodium rushing into the axon. And because sodium is positively charged, you see you're going upwards towards positives, positive sodium ions, positive amount.
So is a threshold also an action potential?
No, that's the beginning of the actual action potential. So from here to here is graded potential. That's a little bit of sodium coming in, raising raising the membrane potential up to -55. When it gets to -55, the chain reaction takes over and no more stimulus is required. You don't have to put your hand on the flush anymore, on the handle. Okay.
At that point, then it's the
At
action
this
potential.
point, the sodium takes over and it's kind of like a brush fire. You don't have to keep adding any more heat. It spreads on its own. So this action potential is kind of like a brush fire. Once you add enough heat, it's going to spread on its own. And all of this above, up and down here, is it kind of going off like a gun being fired or like a toilet being flushed. If you put enough pressure on the trigger, you're going to cause the bullet to come out. And once you start that, the bullet's going to come out. Okay, and takes a certain amount of pressure on the trigger. All right, so in this rising voltage, that's a whole bunch of sodium rushing in. Now remember, sodium was on the outside and potassium was on the inside. So sodium is rushing in. That's causing the membrane potential to go way up. And the potassium isn't going anywhere. It's still staying right where it is. So now we have a whole bunch of sodium on the inside and a whole bunch of potassium making a really high level, +30. Now at this point, you guys see where I am in this point?
Yeah.
This is where the potassium gates start to open and the sodium gates start to close. So because now the potassium is on the inside, it starts rushing out. Now it starts to drop because this is the point where the potassium is leaving. It goes all the way down below rest and overshoots a little bit. Now, the end of the action potential is 5. This is where basically equilibrium. Now you have equal amounts of sodium and potassium on both sides of the membrane, inside and outside of the axon. Now, between 5 and 6, the sodium potassium pumps that I asked you to tell me about on the first test is moving sodium to the outside of the membrane and potassium back to the inside. So getting you back to this point where there's a whole bunch of sodium outside and a whole bunch of potassium inside. So when you get back to rest, it's ready in the same way a toilet has been filling with water. It's ready to have another axe potential. Does that make sense to you guys?
Yeah. Do
Okay,
we need
I
to
know-
know what that phrase is like below the resting point, that hyper,
This is called hyperpolarization.
hyperpolarization? Right? Oh, okay.
That's just because the influx, wait a minute, the outflux of potassium overshoots how it was at rest. And the reason why it's at this particular voltage is because the leakage of it here, when you get all the way to equilibrium, you're actually lower than you were at rest. So this is hyperpolarization. This is the sodium between 5 and 6 is sodium potassium pumps, moving them to opposite sides of the membrane. And when you get back to 6, you're basically back at 1 and you're ready to have another axe potential. If you tell me all of that and a little bit from the book and a little, by the way, would it be a good idea to throw in an analogy into your answer?
Oh, yeah, like your toilet
Always.
flushing.
Right. What 2 things would that tell me? Why would that help you having the analogy?
Because it tells you that we were paying attention to lecture.
That's one. And
And we
second,
get
you wanted to add more detail to your answer, right?
Yeah.
So remember what I said, if you're trying to figure out how little you can write and still get 5 points, you won't. If you say, gosh, I have so much stuff I want to talk about, I'm going to just run out of space. You'll never run out of space. But if you run out of time, you're like, I've said tons and I'm good now. Then you probably get to 5. But if people ask me, Mr. Sage, how long does my essay have to be to get 5 points? And I say, it won't be long enough if that's your attitude. But if you have lots and lots to say and you write a whole bunch, you'll probably get a 5, unless you're just kind of BSing me for half of it. And I can see that too. Don't BS me. Don't write a bunch of nonsense. Throw in analogies, throw in stuff that you've seen in the book, heard me talk about. You should be good to go. All right, got to move on. Okay, so this is another one from chapter 12. Unfortunately, the first 2 essays are from chapter 12. And basically, we did see a little bit of this in chapter 17. Remember, I think, was it 17? When we talked about what happens to cause the muscle to contract, right?
Yeah,
Okay.
17.
So I talked about this already, but here's specifically the figure that shows how it works. So take notes. Now, here we are at the presynaptic neuron. Why do they call it the presynaptic neuron? Because it's on the top side or the front side of the synapse, which is the gap. So I'm going to talk about what happens step by step. Take notes either now or rewatch the lecture.
Well, you said this one is on 12? This picture?
No, this one was, he was talking about the muscle fibers on chapter 10.
Right. So chapter 10, I talked about this process a little bit, but this particular picture wasn't there. But it was more or less showing you the same effect. The only difference was the figure I showed you, what happens down below in terms of what is happening inside the muscle. All I'm asking you with this figure is, how does this mechanism work? So let me tell you, take notes, and then I'll answer your questions. Okay, so the axe potential that we saw in the previous figure is coming down here. It's like spreading like wildfire. It's a chain reaction. So when these gates open, it causes the ones next door to open, and these open the ones next door. So the signal starts coming down to the end. Now, when it gets down here, this figure is not great. But what happens is calcium comes in from the outside and causes these the membrane and dump their neurotransmitters.
Was that the acetyl-CoA?
Right, the acetylcholine. You guys hear me okay?
Yeah, I think you were, well, you cut out a little bit, but from laughter.
Let me repeat.
Okay.
When the signal gets here, calcium channels open, calcium comes in, allows these bubbles full of acetylcholine to dock, dumping the acetylcholine into the synaptic cleft or synapse. When they go onto the postsynaptic side, they cause sodium gates to open here. And then again, that is what is called a receptor potential. The more sodium that starts coming in, the more negative excuse me, the more positive this becomes. So what's happening here is the signal spreading through here. These neurotransmitters are being dumped into the gap. And when they dock over here, the ligand-gated channels with receptors for neurotransmitters, when they stick on that, it allows sodium to come in, and that's the beginning of a receptor potential or a graded potential. That's the term I use, graded potential here, which could cause another AX potential in the postsynaptic or second neuron. Now remember, in chapter 10, this was a muscle cell, but in this figure, it's talking about another neuron that could be stimulated up to a point where you would get another AX potential. If there was enough neurotransmitter dumped, there would be enough of a graded potential generated here to cause an AX potential down below it, moving down the line. Does that make sense? This is not the best figure. I had a much better figure in my other book, but this is the one you guys have in your OER text. So that's what we got. Okay. And one of the things that I do want to tell you about is in the notes for chapter 12, it's going to ask, how are these little circles removed? And it does go into how these neurotransmitters are eliminated. And I'll just give you the heads up. And that is, they either diffuse away, they are absorbed into the bloodstream, or they get broken down into their constituent parts, acetic acid and choline, and moved back up to the top to be reassembled into acetylcholine and put in new synaptic vesicles. So that's the extension. I'm going to ask how you get rid of these, and that is in chapter 12 in the notes. Okay, so there you go. Oh, there's nothing from 17. Okay, well, all right. So you guys get the extra credit here. So I probably should have done 12 and then 17, but all right.
So there's only 2 essay questions?
Only 2 essays, and they both have to do a chapter 12.
Oh, I thought this was chapter 10.
10. Sorry, I'm still thinking of a different book. So chapter 10.
No, they both on a 12.
Everything having to do with neurons.
Is both on a 12.
Essays, and they come directly from your notes in chapter 10, right?
No, 12.
12.
Yeah, both on a 12.
Chapter has to do with neurons, you guys. I thought it was 12.
12.
Yes.
Yeah, it's on a 12. It is on a 12.
Okay,
Yeah.
so these figures are in 12, and I've just talked about them now in detail. So you should be able to generate, both from the notes and from my lecture and from your book, a pretty good essay for each of them. And then the extra credit asks labs to labs. And I'm going to start cracking down on people who start giving me BS answers on this. Oh, the labs to lab was really fun. I learned a lot of stuff, but it's really frustrating because labs to labs can be difficult. That doesn't say anything about the actual lab. You have to do the lab, take notes on what you learned, and that's how you get the extra credit. So I'm going to crack down on this test, whether or not you're just BSing me or you actually did the lab and learned something. Okay, so that's how you get extra credit. It says 0 points because that's how it indicates extra credit and not regular credit.
It
Okay.
could be any lab, right? It doesn't have to be a specific one from this week. It could be any lab.
It can't be one that you already wrote about during test 1.
Okay,
Okay.
thank you.
Any labster that you haven't already discussed as extra credit on the first test. Okay, so now I want to do review, and then we'll do chapter 12, and we'll get through all the stuff that you guys need by the end of today. Okay, let's go to, what's our first chapter? Chapter 12. I know we're putting the cart before the horse a little bit, but let's go ahead and do the review for chapter 12. I believe it's 12A, right?
It's just straight 12. There's no A or B.
Okay, so let's go to chapter 12. Go to your review questions. Were there any in there that you couldn't find or wanted to double check that your answer was correct before the test? And remember, if you have one, you have to read the number of the question so that everybody can find it. We can all do it together. Anybody, chapter 12 review questions. Did you do that?
I have one. It's question number 9, and it's interneurons are found in the, and I just wanted to make sure, the central nervous system?
Yes, central nervous system, correct. Okay, let's do some others from chapter 12. Are there any that you couldn't find or want to double check?
I've got another one. Oh, you can go for it, Erica.
Oh, well, thanks. Number 7, the motor neurons are also known as, was it efferent? Yeah,
Yes,
right?
efferent is correct.
Okay.
Efferent is right.
Let me double
Let's
check.
do some more. More.
Number 12, what is the most common neurotransmitter used between neurons? And I believe it's acetylcholine.
Yes, acetylcholine is correct. By the way, the folks in the chat, I may not have caught all of your questions. There was a question about narcolepsy. I don't know anything about narcolepsy. And this exact picture, by the way, I tried to make sure that every picture that was on the test was also in your notes and in the PowerPoint. Okay, so I should not be asking you about any pictures on the test that aren't in your notes and in your book. Sometimes they're in your notes, but not the book, but I would never ask you to answer an essay of something that was not in your notes. They're always at least in your notes, so you have something to prepare for the test, okay? So John says, what are the 3 major groups of hormones produced by the nervous system? That's a typo, it should be 2. Okay, John, what was the question number?
What number is that?
Yeah, guys, always put the question number so everybody else can find it. What was the question number, John? Hello, John. Ah, he's checking. Yeah, you got to put the question number in so everybody can find that question. He
That
didn't
one
get a
is
number.
from 17B and it's number 18.
Okay, guys, we're on chapter 12 right now. So let's focus on chapter 12 first. So everybody can be looking at the same piece of paper and not jumping between chapters. Okay, so we're in chapter 12. All right, anybody else from chapter 12?
Yes,
review
9 to 26.
Okay,
It's
read it.
potassium gates always open after sodium gates. It's a true or false. Is it true?
Well, did I talk about it just now in the lecture when we were looking at the figure?
Yeah.
Yes, they always open after. So it's always sodium is the rise, and then potassium is the fall, and potassium always happens after sodium. Okay, good.
Can you do 23? Which of the following is not true of a polarized neuron?
So basically what you're trying to find is an incorrect statement. Now,
Right.
often that question doesn't end up on my test because it's kind of nebulous and difficult to answer. But if you know the statements that are true for a polarized neuron, and I throw an incorrect statement in there, that's the right answer because you're looking for the thing that doesn't fit with the others. Okay?
Okay.
If it does happen to end up there, you're looking for a statement that doesn't fit with the other ones, and that's the right. Okay? If you're looking for a statement that is not true for a polarized neuron. Okay?
Oh, how about 21? The
21.
area at which an action potential will initiate if the graded potential is strong enough is called the blank zone.
The
It's
trigger
the trigger zone.
Oh, trigger zone. That's
Trigger
right.
zone, right. Okay, Madison asked, give an example of a place in the body that would have chemical synapses. Is that from chapter 12?
He says it's number 40.
Yes.
40?
Yeah.
Okay, chemical synapses would be the heart. The heart or smooth muscles. Those are both places where there'd be chemical synapses. That was number 40. Okay, anybody else on chapter 12?
Can we do number 5?
5, read it.
It is the sheath that forms an electrical barrier around the axon is composed of, and I'm
Myelin?
not sure if it would be
Myelin.
myelin
myelin.
or glial cells. Would either
Well,
of those count?
Well, myelin is the chemical, and it's made up of glial cells that wrap around the axon. So the glial cells contain the myelin, but I was going for the chemical there. So the answer I'm looking for is myelin.
Okay. All right.
Anybody else, chapter 12?
28.
28, read it.
Yeah. What terms refers to the state of a neuron immediately after the potassium has diffused out? Would it be repolarization, or would it be sneak peek?
It should be
Or is it hyper?
hyperpolarization. Hyperpolarization is what I'm thinking. After it's diffused out. I think what I was going for there was hyperpolarization. Is that what your notes say?
Yeah, well, because all I know from what you said is it has to go through hyperpolarization before it can get to repolarization. Okay.
That's it. And if for some reason you missed that and you want to go through that question with me next week after the lecture, and you say, hey, Mr. Sage, you said, and you owe me a point, I'll happily give it to you, okay? if for some reason that's wrong and what I said just now was incorrect. But in my memory, it's hyperpolarization. Okay. Anybody else from chapter 12, please?
Number 34.
34.
The number of action potentials per second is called the
Frequency.
Frequency. Dang, okay.
Anything that's number per second is always going to be frequency. And here's the thing, you guys, let me add a little bit to that. Your body codes by how intense the stimulus is by how many action potentials come through at a particular time, or the frequency of action potentials. One of the review questions says a really strong stimulus will cause a more intense action potential, and that's false. Action potentials are always 100 millivolts total, or from -70 up to 30. And the difference between a strong stimulus and a weak stimulus is not how strong the action potential is, but how many come per second. So a strong stimulus is going to be a lot of signals coming through quickly, and a weaker stimulus is going to be fewer coming. So lower frequency, lower stimulus, faster frequency, greater stimulus. Okay? Hopefully that'll help you when you go to take the test before Sunday, right? Take the test before Sunday.
Oh, can we do one more on 38?
Okay, go. 38. Read it.
How does Novocaine cause numbness in a nerve?
Okay, so the section of the notes was called cane drugs, and what it does is it blocks the ion channels. So because sodium and potassium can't move, prevents the signal from getting from your, let's say, your mouth to your brain, so you can't feel the pain.
Okay.
And eventually it diffuses away and you can start feeling it. Okay.
I have a question before you move on. My computer froze when you were giving the explanation for answer 34.
What was 34 again?
Oh, it was frequency. The number of
Frequency.
action potentials per second.
The
Thank
number
you.
of action potential per second is called frequency.
Thank you very much.
You're welcome. Want to make sure everybody gets what they need. Any others from chapter 12? I think pretty soon we got to move on to our other chapter, which is 17.
We did 17.
17 we are done, so probably 10.
10.
Okay.
We
So
haven't
let's do
done
a
the
couple
review
more
for
on
17.
12. Let's do a couple more on 12 and then we'll move on to 10.
10 is for the following week though, isn't it? In terms of the exam. So for this week, it's 17 and 12.
Yeah, 17 and 12. So we've only done
12.
right? So we need to do 17. Yeah.
Don't we need to? Well, okay.
Well, look.
I mean, I know we didn't actually
chapters that
go
are on
through
this
12,
week's
even
test.
though we're answering the review questions on 12. You know what I'm
Wait, say
saying?
that again?
We didn't actually go through 12 with your PowerPoint, but we did,
Right.
even though we're doing the review questions.
Right, because theoretically you guys filled in the notes, you worked
Yeah.
on the review questions, and then whoever wants to stick around can get the cart after the horse or before the horse or whatever. I wanted to make sure people got their review questions done, even though I haven't lectured on it yet. There may be times this semester where I won't be able to lecture on a particular chapter, but I'll try to lecture on most of them. At least you have the notes. And remember, the test questions come from the review comes from the notes. So once you get those notes filled in, you have all the most important information that you need to take the test. Okay?
Will we be reviewing the first two chapters that we went over at the very beginning of your lecture today before the
that'll be
exam
next
next week?
That's next week. So it'll give you time to get your review questions done. And so when I see you next Tuesday, those should all be filled in, and you should have the ones that you're uncertain of marked so that we can go through quickly and get those answered for you. Okay. I just didn't want to get any further behind. I wanted to do what was on the docket for today, at least part of it, and then play catch up at the end. I probably should have done it in reverse, but it is what it is. And look, you guys, for an online class, you don't always get a lecture, do you? Right? Sometimes it's just, here's the PowerPoint and here's the test, and that's all you get. So some is better than none, right?
That sure is.
Okay. I know people want what they want, and I want to make you guys happy, but I can only do so much. And yeah, I'm only going to lecture once a week. We're going to do the best we can with once a week. Anybody else for chapter 12? Okay, not hearing
Can we
anyone.
do 27?
27, last one. Let's go, read it.
The strength of an action potential is dependent on the strength of the grade potential, which has triggered it. Is
Yes.
that false?
That's false. Remember, I said action potentials are always 100 millivolts, regardless of, remember, you only have to get past from graded potential to the above the threshold, and then you get a full action potential. So the strength of the action potential is not dependent on the graded potential. The graded potential just has to get above threshold. And once the graded potential goes above farther above threshold, the action potential doesn't get any stronger. You just get more of them. Does that make sense? So it's
Yes.
number, not intensity. Okay, so that's a false statement. All right, guys, we have limited time and I got stuff I got to go do. So let's go to, what is it, 10? This is the chapter we haven't covered yet.
17A for
17A.
review.
17A. Okay, sorry, guys. These new chapter numbers are throwing me off. 17A. Let's start. Who's got something from 17A?
Number 19.
19, read it.
Why must steroid hormones be transported by carrier protein?
Okay, what did I talk about today?
Is that the water soluble or
Right, because
in
they're
oil?
water
Okay.
insoluble,
Okay.
and they can't travel through the watery blood without a transporter protein.
Thank
What
you.
number was that again?
19.
Oh,
What was the number?
19.
19. Hey, folks, when somebody announces the number, try to find that quickly rather than looking through your own notes. As soon as somebody says it, stop what you're doing, find their question, because then otherwise you have to ask what was the number again. So
I was still trying to grab that chapter when
That's
we
okay.
switched over.
yeah, I know. There's a lot of stuff happening really quickly. We're doing the best we can. Good job to all of you for being here and participating. Would somebody else like to do another question from this chapter?
Can we do the one from the chat? It's number 15. What is a cascade reaction?
A cascade reaction, that one's kind of like nebulous. It doesn't have a clear answer, but just in case it ends up on the test, a cascade reaction is where one reaction leads to another, to a third, and then to a fourth. So it's a series of chemical reactions dependent on each other. That's what a cascade reaction is.
could you
Okay.
summarize that as a domino effect?
Yes, kind of like a domino effect. I mean, that's an analogy, not a summary.
So you have to literally say chemical reaction that leads to one chemical reaction and then another.
Well, I mean, it's multiple choice, you guys. If it ends up on there and you see something like that, then pick it, right? I'm not going to ask you to tell me what a cascade reaction is, because the only time you have to tell me in sentences are the essays. Does that make sense? We don't have short answer on my tests. They're either full essays or multiple choice, true, false. Okay,
Number 2.
2, read it.
When the effect of a hormone reduces a stimulus, this is called.
Okay, what was the really super important thing that I talked about today in terms of when the hormone does get you back to normal and now you don't need to do anything else? Do you remember that?
Negative feedback.
Negative
Negative
feedback.
Oh,
feedback.
negative feedback. Okay.
Negative feedback is the foundation of homeostasis, and homeostasis is the foundation of physiology. So really, you could say negative feedback is the foundation of physiology. Most physiology is based on negative feedback.
Can I ask for number 8? Can I do number 8?
8, read it.
What is the problem with talking about human pheromone activity?
Okay, that one is on the test. So what is the problem with talking about human pheromones?
There's
What
no
was-
solid research on it that can.
Right, we
really
don't know
identify it.
exactly which ones, whether or not we have them and which ones they are, okay? Because we're not, by the way, it is on the test, and one of the wrong answers is it's very hard to pronounce, so don't pick that one, you guys.
Thank you.
If you remember my lecture today, you'll remember that we're just not sure exactly whether or not we have them, and if so, which ones. So that's the answer there. Okay, more, please, on this chapter.
Number 6, when a molecule has a direct effect on DNA, it has a what? And I think it's genomic effect.
Genomic, and that goes directly to one of the essays, right? By the way, be sure to use the term genomic effect when you're, wait a minute. That's not on this test, is it?
No.
No. All right, well, it's multiple choice.
Thank you for
took
giving
that
us heads
essay
up
out.
for the future test, though.
Maybe, but I took that essay out because that figure that showed the genomic effect was pretty weak, and it just didn't have enough detail in it to show the genomic effect. I probably should have put it in there, but anyway, you guys are probably happy there's fewer essays. Some people actually really like the essays because they shine when they write. Some people hate them. So I think 2 is fair for this test.
Can we do 11?
Go.
The protein-based hormone that cannot enter cells is called,
Peptide.
oh, was it peptide?
Your eye open.
What was it?
Peptide hormones or amine hormones both might be correct answers there. So keep your eye open for a choice that gives you multiple right answers.
What if it has both of
Bye,
them?
John. Have a good one. Yes?
If it, is there a possibility for that question to have both amine and peptide on there?
Yeah. So if you see a choice that says 2 of these answers are correct, what are you going to do?
Oh, well, select that one. Okay, but
Yeah.
I just didn't know.
Always select the choice that gives you multiple answers unless you're certain one of them is completely and totally wrong. Okay? That's the only time you don't pick all of these are right or 2 of these are right. When I offer that option, 95% of the time that's the right answer. So please try to remember that on the test. When you see an option that says 2 of these are right or all of these are right, you need to have a really good reason not to pick it. Okay.
Speaking of that option, going back to the exam that we just took, there was a question that had something to do about, I want to say,
I know which one you're talking about. That was one of the 5% where the all of the above was wrong because one of the choices was the opposite of the right answer.
right, okay. I was just
Yep.
double checking to make sure that was one that I got right.
Yeah, no, you did. I mean, if you rejected that all of these are right, I just don't want people picking it every single time and say, oh, he just told us to pick it every time we see it, never even look at the choices. So even 90 to 95%, if you never did look at it, you'd still get an A's worth of points on it. But on that particular one, it was a distractor, which means it wasn't the right answer. Because one of the options was completely opposite of the correct answer.
Membrane thickness, etc.
Yeah, because membrane thickness, you don't want that going up to increase diffusion. You want it going down. So that one didn't go with the other ones. So yeah, so it was just, I want to say just one right answer on that one. Look, you guys, these tests can be a little bit confusing, but because you have your notes with you, I'm asking you to do a little bit more critical thinking. And if there is a problem with the test, come to lecture, talk to me about it, and we can hammer out whether or not you deserve the point. Okay? Anybody else for this chapter that we're in right now?
Number 12. An input or signal that begins a reflex is called a stimulus.
Go ahead.
It's number 12. An input or signal that begins a reflex is called a stimulus.
Stimulus is correct. Very good. Okay. Anybody else for this chapter?
Number
Nobody?
5.
5, go.
Which of the following is not a way that hormones works on its target cell?
Incorrect would be the right answer. You're looking for something that doesn't match that section of the notes.
Okay.
Yeah, I realize there's no like clear answer there, but if you know the ones that are true, then one that stands out away from it would be the correct answer. It's not one of the ways that it works. So just know the ways that it works. And then if I throw in something that's wrong, that's what you're looking for, something incorrect. Anybody else? All right, so folks, we're going to our last phase of today's lecture, which is anybody who wants to stick around in office hours and talk about test 1 and have me go through it with you and we can discuss the reasons why you missed particular questions. Does anybody want to stick around for that?
I would like to, yes, but are we also going to do 17B?
Thank you, Elizabeth.
17B, I'm going to try, was that one on the test or for next week?
No,
This
that's
week.
This week.
That was this week. Okay, I'm going to try to play catch up on Tuesday for 17B. Okay,
No, it's for this
That's
week's
for
exam.
the exam.
it is this week's exam.
Yeah, 17A, B, and 12 are for this week.
We just need to do the review questions for 17B.
Okay, let's do, okay, so let's do 17B now, please. 17B review questions. Can you guys pull those up?
I got it.
I have a question on number 7.
Go for it.
What do hypothalamic neurohormones control?
And wait, this is on this week's test?
Yeah,
Yeah.
Yep.
Okay.
17A and B and 12.
you can take it off if you want. We don't mind.
No, the test is already set. The test is already set. Okay, read the question again, please.
What do hypothalamic neurohormones control?
The pituitary gland.
Okay.
Yeah. Okay.
I had a question.
No.
Question 14 as well as a couple more.
Well, let's just start with 14.
Okay, they make a reference to melanin.
Oh,
They
that's
say-
a typo. It should say melatonin. Sorry
Okay.
about that.
So that
So,
would
yeah.
be, yeah.
Melatonin. Please change that to melatonin.
Oh.
I just saw Dakota White just now sitting in the waiting room. Okay, anybody else for 17B?
Number 12, what is
Oh.
SADD and what are its treatments? And I know it's seasonal affective depressive disorder.
I
but I
forgot
don't
to
know about
talk
the
about that.
treatments.
The
Yeah.
treatments are artificial sunlight. You get it in winter months in places like Montana and Alaska. It can cause depression. And the treatment is to get artificial sunlight shining on your face so that you get your
So
eyes.
like
stimulated.
sitting in a tanning bed or something?
Yeah, even that would work.
horrible.
But just even a sun lamp on your face is enough to cure you of seasonal affective depressive disorder. I forgot to talk about that today. Okay. Anybody else? 17B.
16.
16, read it.
The posterior pituitary synthesizes the hormones that it releases. Would it be false?
False.
Okay.
Yeah. Casey asked question 4. What are 3 hormones that can all target a single cell or tissue? And my brain is blanking. It was somatomedins. You guys have that one, number 4. Who's got number 4?
Oh,
Wait, which one are we on?
the answer.
It's 17B and it's number 4.
What are 3 hormones that can all target a single cell or tissue?
Was it cortisol and glucagon and epinephrine?
Thyroxine, growth hormone.
Yeah, thyroxine, growth hormone, and
Melatonin.
oh,
Oh, wait, shoot. Where is
No,
that?
the somatomedins.
Oh, adrenal
somatomedins are one of them. Where's the
That's a
section?
heck of a-
Somatomedins, guys?
Okay, I'm confused with that one. So what's the answer for that?
I can't remember. You guys got to find the section of your notes where it mentions somatomedins.
It's somatomedins, thyroxine, growth hormone.
Right. Okay, here it is. Growth hormone, somatomedins, and thyroxine.
Oh,
That's
I
it.
see.
Okay. I know it's a little confusing, but all right. Any others for this chapter?
So
Number
for
34, sorry.
No, it's okay. Go ahead.
Go ahead,
what affects-
Erica. Wait, who's going?
Go ahead for 34. Whoever wants to get there.
What effects does melanin have on the body?
Should be melatonin.
Okay, just wanted to clarify. Thank you.
Read it.
And it's antioxidant effect, right?
Well, you got to read the question. I don't have the questions.
What effect does melatonin have on the body?
It makes you sleepy.
Oh, it makes you sleepy. What about the antioxidant effect?
Yeah, there's a question that says melatonin has antioxidant effects similar to what, or something like that. It's vitamin C. I can't remember what the
No, the
question
question is, what effects does melanin have on the body? Or melatonin is what it's supposed to be.
It
But
should be melatonin, and the answer is makes you sleepy.
Okay.
It's a typo. It should say melatonin, not melanin, because we never talked about melanin in this chapter.
So for number 9, it says the growth hormone, somatomedins, and thyroxine all have the effect of increasing, is it IGFs?
Hey guys, I have some news. My kid is stuck at the bus stop and I was supposed to be there 20 minutes ago to pick him up. So I got a jet.
Okay.
I'm sorry, you guys. We'll talk more about your previous test next week. I'm sorry to cut you guys off, but I gotta go pick him up. So I gotta sign off. Hey, good luck with this week's test. I'm sorry I don't have more time.
Can we
And,
email you the questions that we have the answers to, just to see if they're correct, if that's possible?
I have to leave right now, you guys. I'm leaving. So I'm just saying goodbye. I'll see you guys next Tuesday. And anything that you have more, we'll have to do on next Tuesday. Okay. I gotta go. I'm late picking someone up. So, okay. Have a good week.
Thank you.
Good luck with the test. And I'll see you guys on
Thank you.
Tuesday. Bye guys.